A very good morning, everyone. I welcome you all to the second Pharma Summit, Drug Discovery and Community Trial on 4th and 5th December 2023, a hybrid conference organized by Association of Pharmaceutical Research, APR. About APR, Association of Pharmaceutical Research is one of the world leading professional association for pharmaceutical and life science professionals caring for people. APR is a non-profitable professional association meant for research and development in the field of pharmaceutical science and technology. APR is an international forum for researchers, academicians, doctors, and practitioners for sharing knowledge and innovation in the field of pharmaceutical science and technology. APR aims to bring together worldwide researchers and professionals, encourage intellectual development, and providing opportunities for networking and collaboration. APR meets with its objectives through academic networking, meetings, conferences, projects, research publications, academic awards, and scholarships. APR strives to enrich from its diverse group of advisory members. APR connects pharmaceutical researchers and academicians by organizing international conferences, world conferences, faculty development programs, international workshops, seminars, guest lectures, short term training programs, providing memberships, establishing chapters, faculty exchange program, and in-plant training. About BioLix. BioLix is a not-for-profit professional association which prominently promotes research and development. We at BioLix have brought a revolution in the field of worldwide conferences. BioLix conference bring together the professional wizards and leaders who have explored all avenues to reinforce the field of life sciences and medicine technology. BioLix conduct events worldwide which help in enhancing the skill of the people from diverse industries and forms a common platform for eminent personalities, physicians, researchers, doctors, and academicians, and professionals, business figures, and much more. About our academic partners, Chettinad Academy of Research and Education (CAP) was conferred the status of deemed to be University of the UGC Act 1956 in the year 2008, and since then it has been contributing significantly to the society as an institute for higher learning and research in the field of medicine, allied health sciences, and nursing. The mission of the School of Pharmaceutical Sciences is to be a national center of excellence in preparing graduates for a lifelong professional career in professional care that is tuned with the need of society. The school provides an excellent learning environment conducive to the pursuit of professional competence while providing services to the community and undertaking relevant translational research for the enhancement of health. The school will also endeavor to establish training programs in the field of pharmaceutical sciences, lending support to the human resource development initiatives of the country. Adichum Chanakari University is a professional research oriented, student centric, multidisciplinary, not for profit state private university. Within the short period of its existence, it has emerged as a national renowned higher education university. The university has a host of institutes, departments, and centers, including medicine, pharmacy, nursing, and engineering, management, commerce, and education. Sri Adichinga Chanagari College of Pharmacy, a constituent institution of Adichinga University, is one of the leading institutions in the country, offering pharmaceutical education at the undergraduate, postgraduate, and doctoral level. Sri Adichinga Chanagari College of Pharmacy was established in the year 1981 and running D Pharm, B Pharm, M Pharm, Pharm D, and PhD programs. It is approved by AICTE and PCI New Delhi. The college operates with a mission to produce globally competent pharmacists with skill, knowledge, and attitude. While the institute's mission is to impart quality education with training to excel in the pharmacy profession. About the conference, Association of Pharmaceutical Research (APR) organizes the second Pharma Summit Drug Discovery and Community Trial on 4th and 5th December 2023. This conference aims to bring together leading academic scientists, researchers, and scholars to exchange and share their works and research results on all aspects of pharmacology. Pharmaceutical biotechnology, pharmacogenomics, drug delivery, biodrugs, pharmacovigilance and drug safety, pharmaceutical microbiology, pharmaceutical research and development, pharmaceutical analysis and quality curatives, biotherapeutics, radiopharmaceuticals, vaccine design, formulation technologies, clinical pharmacy, industrial pharmacy, pharmaceutical chemistry, and pharmaceutics. The drug discovery industry has become such a competitive market that it continually faces a challenge to find better drug delivery technologies. This industry but must discover and develop innovative drugs for a wide range of diseases in a marketplace that is likely to experience growing regulatory challenges, pricing pressures, and various other bottlenecks. Pharma Summit 2023 will be welcoming brainstorming research papers from across the world in the field of pharmaceutical science and drug discovery. I would like to welcome all our honorable dignitaries of the Day 1. Our conference chair, Dr. Anil Philip, Associate Dean, School of Pharmacy, University of Niswa, Oman. Dr. M. Sridhar R., Vice Chancellor, Chettinad Academy of Research and Education, Chennai, India. Mr. Rudra Banu Satpathy, Founder and CEO, Association of Pharmaceutical Research and Technored Group, India. Dr. B. Ramesh, Dean, Academics and Accreditation, Adichunga Chanakari University, India. Our keynote speakers of the day, Professor Dr. Pramod Kumar Rajput, MBA, PhD, FIA, SSD, FIA, ECT, FGCPR, USA, MLE, Life, Member of Leadership Excellence, Harvard Square, Cambridge, MA, USA, and the Indian Society for Training and Development, India. Dr. Nahla Elkotsha Ismail, Professor and Dean, Bursar and Council Member, Malaysian Academy of Pharmacy, Department of Clinician Pharmacy Practice and Pharmaceutics, AIMST, Malaysia. Dr. Professor Lakshmi, Professor and Dean, Chettinad School of Pharmaceutical Sciences, Member Board of Management, Chettinad Academy of Research and Education, India. I would like to welcome our session chairs of the day, Dr. Batma M. Parag, Principal, Department of Pharmacology, the Oxford College of Pharmacy, India. Dr. S. Murugesan, Associate Professor, Nanjumaruthvam, Siddha Toxicology, National Institute of Siddha, India. Dr. Devi T. Professor, Pharmaceutical Chemistry and Analysis, Chichenad School of Pharmaceutical Sciences, India. Dr. Rokia Sultana, Professor and Vice Principal, Eneopa Anipoya Pharmacy College and Research Center, India. Dr. A. L. Agilandeshwari, Associate Professor, Chettinad School of Pharmaceutical Sciences, India. Dr. P. Muralidharan, Professor, Department of Pharmacology, Chettinad School of Pharmaceutical Sciences, India. Assistant Professor Ravi Nandan, Assistant Professor, Department of Pharmacy Practice, Sri Siddhaganga College of Pharmacy, India. And I welcome our delegates to this 2 days of event. I would like to welcome our first keynote speaker of the day, Dr. Pramod Kumar Rajput, MBA, PhD, FIA, SSD, FIA, ECT, FGCPR, USA, MLA, Life Member of Leadership Excellence, Harvard Square, Cambridge, MA, USA. The Indian Society for Training and Development, India, about him. About him, a global speaker, international life coach, mentor, corporate trainer, educator, thought leader, author, changemaker, chief guest, host of honor, keynote speaker, moderator, and active panelist on numerous national and international forums and events, along with 13 honorary doctorate in various segments. He has a rich experience of more than 40 plus years in the pharmaceutical and healthcare industry in sales and marketing as senior vice president while taking care of entire India and 25 countries. With a great passion for education, a blend of business building added with the strength of education based on skills and knowledge. He is a visiting professor in many universities and also on the honorary board of various institutions and organizations. I welcome our first keynote speaker of the day, Dr. Pramod Kumar Rajput, to give a keynote address. Sir, sir, we can't hear you, sir.
Fine is okay, fine. Thank you so much. And very, very good morning and good afternoon to all the wonderful, eminent people who have joined here on
Good
this
morning.
second important summit, which is happening here in this particular hybrid mode, right? And thank you, Nivita, for such a great introduction of APR about the Chinchagiri College. I was very happy. I was feeling
Thank you, sir.
overwhelmed. Yeah, I'm feeling overwhelmed that I'm a part of APR group, right? And I'm a member where I keep on learning first and then keep on sharing. Because for me or everybody, learning is the first motto and then sharing is the second motto. When I have been invited, being a part of sales and marketing, being a part of pharmaceutical industry, I thought that I'm very blessed because I will be able to talk to the all the young minds along with the all professors, great professors, which I'm able to see because the professors are the torch bearers for our nation, and the young generations, they are the star for the entire future. So this talk is based on one of the very wonderful subject. The subject is on the drug discovery and community trial. This is the real requirement. Today, Bharat is known as the second pharmacy of the world. And let me tell you, we are the number sixth in terms of providing the GDP for our country, while providing a lot of the drugs, what are the trials and all. At the same time, the new discoveries are happening. So I would like to add it here as a keynote speaker, the significance of the today's topic, the drug discovery and community trials, in revolutionizing the healthcare segment and improving patient outcomes plays a pivotal role in revolutionizing the healthcare and improving the patient outcomes both together because they go hand in hand. And there are some of the points which I'll be talking about it, that is addressing the unmet medical needs, which are very essential for finding effective treatment for various diseases. Personalized medicine, which we call as precision medicine, the drug discovery, which allows for the development of the personalized medicine, accelerating the innovation while providing a platform for collaboration between scientists, clinicians, patient and industry partners, patient engagement and empowerment, bridging the gap between the research and the practice, which is the need of the hour, and health equity and access to the care. So my talk will be on the parameter, which is the power of drug discovery, power of community trials, promoting diversity and inclusion in the clinical trials, which are important, how to have ethical consideration and the patient-centric approach, and what are the future direction and the challenges. So the point one is, what is the power of drug discovery? Because it lies in its ability to transform the healthcare and improve the patient outcomes through innovative treatments. Because the drug discovery enables the development of innovative treatments that target specific diseases, pathways, or mechanisms, which leads to the discovery of the new drug that can effectively treat previously untreatable condition or improve the existing treatment option. Both. That is the requirement of the innovative therapy, innovative treatment. Second is the disease prevention and the management, because the drug discovery plays a vital role in identifying preventive measures and managing the strategies for various diseases. So that is a part of the disease prevention and management. It is going to help, the power is going to help for the improved patient outcomes, as it gives a significant improvement in the patient outcomes by developing effective treatment and therapies. Because drug discovery can alleviate symptoms and show disease progression. Then it comes to the advancement in research and technology. Today, that's what we are going to speak on this part, because drug discovery drives all the advancement which is required in the research and the technology, because it pushes the boundaries beyond the limit of, when I say the beyond the limit, it is of the scientific knowledge, encourages collaboration between the researchers and industry partners and educators, because today we are here in front of the educators, the senior members from across the globe, and the researchers which are here who are listening this meeting, and industry partners who are also a part of it, which is going to foster the development of the cutting edge technologies and methodologies that enhance the drug discovery process. Of course, it is going to take care for the economic and societal impact, because when the better drug is going to happen and when the two-ways condition is going to take care, it is going to increase the economic condition of our India, not only of the India, for the entire universe, and societal impact. This is the power of the drug discovery. Now, the second point comes about what is the power of community trials. When we talk about the community trials, because it is empowering the patient and communities both, because here we have to have the patient-centric approach. Why? Because the patient-centric approach, it leads to the development of the treatment that are more effective, accessible, and aligned with the patient values. That is the point number one, which is the power of the community trial. Then it also improves the treatment outcomes, because community trials provide a wide opportunity to evaluate the real world effectiveness of the new treatment, because by involving diverse patient population and considering the factors such as age, gender, ethnicity, and socio-economic status, these trials generate data and that better reflect the broader patient population. It also enhances the access to the clinical trial for underrepresented population. I repeat, for underrepresented population. I'm not talking only about India. In the entire universe, there are many underrepresented populations are there. So it is going to help about it. It also helps the empowering the patient and the communities. It also helps for addressing the health disparities. How? By actively involving these communities, trials can identify and address barriers to the care, develop culturally appropriate intervention, and thus it ensures the equitable access to the new treatment. Then the last point is in the power of community is building the stronger healthcare system, because very soon our India is going to be the number one in the pharmaceutical industry, in the healthcare system, in clinical trial. You talk anything, we are going to rule the roost. When we are going to rule the roost, it is required, because with this, we are able to foster the collaboration between the researchers, healthcare providers, and community organization. Why? Because it promotes the knowledge sharing. Today, we are sharing the knowledge. Everywhere we talk about the same thing. Capacity building, which is the requirement by the industry people, and the development of sustainable healthcare solutions that are response to the need of the community. So in that way, we will be able to take care. Such an important part is the power of the community trial. The next point I would like to have, why we should promote the diversity and inclusion. Diversity and inclusion in the clinical trial, because it is highly essential to ensure that the research findings are applicable to a wide range of individuals and to address the health disparities, because a lot of the disparities are happening. And it can be taken care with a couple of the important points, like define inclusive eligibility criteria. Define inclusive eligibility criteria. Because when we define the inclusive eligibility criteria, it should be, it is going to include a diverse range of participants. And this includes considering factors such as age, gender, race, ethnicity, socioeconomic status, and comorbidities also. Second point I like to add here, it also engage underrepresented communities, which I spoke on my power of the trials. So I will not repeat the same thing. At the same time, it increases the access to the clinical trials, because while increasing the access of the clinical trials, it brings the trials closer to the potential participants. Geographical barriers can be reduced and making it easier for individuals from the diverse background to participate. Next important point I like to highlight it here, it addresses the language and the cultural barriers, because there are a lot of barriers are there which are becoming an hurdle, but we are able to take care for this. It translates the study materials, offering interpretation services, and it ensure the cultural sensitivity in the trial procedure, which is basically required. This also helps the educate the and train researchers, because if in this way, when we are able to take care for the cultural barriers, address language, we'll be able to take care for this important segment which is required in the healthcare industry. It collaborates also in community stakeholder, because that is going to lead about the collaboration and community stakeholders. It promotes also the diversity in the trial design. So these are the important things and area, and at the same time it helps in the addressing the financial barrier, because many times such clinical activity which we are talking about it, which is the basic caption of this conference, is that it is going to take care. Now how to have this ethical consideration and patient-centric approach, as it is a crucial in the healthcare and clinical trial to ensure the well-being of the right of the patient. I repeat, the well-being of the rights of the patient, which is very important. And when we want to talk about the well-being of the rights of the patient, the couple of very important points which comes in the entire segmentation, which is respect of the autonomy. Because if we respect the autonomy, which is a fundamental right, it involves providing patient with comprehensive information about the trial, including the potential risk and the benefit. So the patient will not be in the dark, and patient will be very clear what he is there or what she is there, and thus obtaining their informed consent before participation. So in that way, the trial will be very smooth. There will no hurdles will be there. Second is the beneficence and the non-maleficence, because the principle of beneficence, which we call is acting in the best interest of the patient, and non-beneficence, which we call is that do no harm. No harm is there. It guides the ethical decision making. Privacy and confidentiality is going to be maintained, because it is very essential. This is a basic requirement of the ethical consideration. So patient data should be handled securely and in compliance with the relevant privacy regulation to maintain trust and confidentiality. Patient-centric approach is going to increase with the privacy and the confidentiality. Shared the decision making will be another very important part is going to happen, because when we talk about the shared decision making, it is an ethical approach that involves collaborate decision making between the patient and the healthcare profession. Why? Because it ensures that patients are actively involved in the decisions about their care, including participation in the clinical trial. Because what is ultimate motto? That the trial should be very smooth. Transparency and accountability will be maintained, and continuous ethical review will be the requirement, because it is going to happen. So there will not be any heartburn for the continuous ethical review, because everything will be very, very smooth, and which is the real need of the hour. So what are the future direction? Then when we talk about so many things, then there has to be the future direction and challenges, because now artificial intelligence is coming up. We are talking about the AR, VR. We are talking about the complete change. So the field of the healthcare and the clinical trials is constantly evolving, and there are several future direction and challenges to consider. And this I'm not talking about only from the India per se. I'm talking about the entire globe, what the entire universe, entire industry is going to do it. And that is advancement in precision medicine. That's what the first thing is there, because precision medicine, right, aims to tailor medical treatments to individual patient based on their unique characteristics, genetics, and environmental factors. That's why we said about it, precision medicine in a layman language is known as a personalized medicine. And the future of the clinical trials involves incorporating precision medicine approaches to develop the targeted therapy and improve patient outcomes. Because now everywhere it is going to be a niche therapy, niche requirement. The healthcare professionals will be very, very niche about it. The segmentation is going to happen, right? In the MBA, we read about STP, segmentation, targeting, and the process. Same thing is going to happen in this healthcare industry, and that's why the researchers are working about it. Integration of the digital health technology, the integration of digital health technology such as wearable devices, remote monitoring, and the telemedicines presents new opportunities for the clinical trials, because now every second person, third person is using the wearable devices, and that is the basic thing is there for the entire globe. inclusion of underrepresented population, because ensuring that diversity and inclusion in clinical trials remains a challenge. Future direction involves actively addressing barriers to participation for underrepresented population, which includes the racial and ethnic minorities, women, children, and older adults. Next point I like to highlight it here, patient-reported outcomes and patient engagement, because the future of the clinical trials emphasizes the importance of patient-reported outcomes and patient engagement, because PRO, which we call it, it captures the patient's perspective of their symptoms, quality of life, and treatment experiences. Next point is big data. Big data is the real requirement, and AI, artificial intelligence. The use of the big data and AI in the clinical trials, it holds a great potential. Analyzing the large data sets can provide insight into the treatment effectiveness, which is the need of the hour. Identify the subgroups of the patient, which is again the need of the hour, who may benefit from the specific intervention, and optimize the trial design and recruitment strategies. However, the challenges related to the data privacy, data quality, and algorithm needs to be addressed, because this is the real fulcrum to have the real outcome from the big data and analysis. And that's why AI is the role is coming up. Ethical consideration in emerging technologies is again coming in a very big way. Global collaboration and data sharing, because collaborative efforts and data sharing among the researchers, institutions, right? And today we have the researchers here, we have the institutions here, and regulatory bodies. I'm sure regulatory bodies would have joined. They all can accelerate the pace of the clinical research, because the future direction involves fostering the global collaboration, harmonizing the regulatory standard, and promoting data sharing while addressing the various concerns which are coming up, the various hurdles which are coming up, the various hiccups which are coming up related to the data privacy and intellectual property. Regulatory and policy frameworks can also be taken care very well, which ensure the patient safety and adapting to the changing landscape of the healthcare and the research industries. So for the conclusion, I would like to highlight it here, how we can inspire the audience, how we can inspire the entire people who are listening, who are the researchers, who are the institutions here. It is the point number one is leadership and empowerment, because strong leadership is essential to inspire and drive the change, because change is required and we have to change ourselves as per the recent scenario, right? What are the abilities which we have, and what are the various things which are available in the entire market. Leaders should actively involve themselves in the healthcare initiative, demonstrate commitment, and empower staff and community member to participate. And that is the role of the leader is coming up. Leader cannot be making himself or herself at one side. Leader has to lead the thing to make the things happen. Education and awareness. I'm very happy that today the great educators are there. They are going to give their knowledge. They are going to share the knowledge, because they are a part of the pharmaceutical industry. And while increasing their wonderful knowledge, the recent knowledge about the importance of the healthcare initiative and their wonderful potential, they are going to impact is in a very, very big way, because educating the audience about the benefits of supporting these initiatives, such as improved patient outcomes, access to innovative treatments, and advancement in the medical research is the need of the hour. Engaging the community, we are going to engage the community in this way. We are able to take care for the diversity and inclusion, which is the need of the hour. We are also going to take care for the patient-centric approach, which I just now spoken in my previous 10 minutes before talk. It also is going to help about the utilizing the digital health technologies, because here I like to highlight it here that the leverage the power of digital healthcare technologies to engage and empower individual, because now the world is changing totally. We have to depend on the digital health technologies, and this is the future which is there, because 2030 we will see that how the things are going to be fasting, moving about the things. The trial which you used to have 3 years to 7 years of time, in 6 months of the time the trial will be over, and we are going to provide the personalized medicine, precision medicines to the patient which is required. Highlighting the success story, we should be blowing the success story. We should be talking about the success story, which is very important in the advancement of the healthcare and research, because every person who is being a part of the clinical advancement and research, he or she should be acknowledged for the smallest thing, and that gives us the encouragement to do, let me make more. Collaboration and partnership is the need of the hour. Today, there is no competition. Today, there is an healthy competition. When we talk about the healthy combination, it is a part of the collaboration and the partnership. The better we collaborate, the better we have the partnership, the more we learn, because it is like a collaboration of the multiple brains. We are going to foster the difference of the stakeholders, including healthcare providers, researchers, policymakers, industry leaders, and the patient advocacy groups. So encouraging the partnership that promotes knowledge sharing, research policing, and collective action to drive advancement in the healthcare. And two more points it is coming in my mind, which is the ethical consideration. Emphasize the importance of ethical consideration in healthcare initiative, because that is a long-term process. We cannot have the shortcut. So addressing any concern or misconceptions related to ethical issues to build trust and support is going to help to have a long-term benefit, and then we'll come to the result which is required for the goal which we have decided. Measure and communicate the impact. So in this way, these are the couple of the suggestions are there which I thought on this wonderful conference we can talk, and we can highlight about it as a keynote speaker in front of such a great people, 70 people, in front of all the great educators, the dean from the different places, and the research which are here. So thank you so much for giving me a great opportunity on this hybrid mode, and I once again feel happy and blessed that I'm a part of APR, which always keeps on building the community to build the entire nation to take care for the universe. Thank you so much once again, APR and Nivedita. Thank you.
Thank you, sir, for your informative session. Thank you so much, sir.
Thank you so much. Thank you so much.
Thank you, sir. Now, I would like to welcome our conference chair, Dr. Anil Philip, Associate Dean, School of Pharmacy, University of Nizwa, Oman, to deliver the welcome address.
Participants, I am thrilled to welcome you all to the second Pharma Summit on Drug Discovery and Community Trials to be held in the vibrant city of Dubai, UAE. As we gather for this prestigious conference, we welcome the diverse community of pharma students, esteemed faculties, and the visionary pharma industrialists. Together, we will explore the latest breakthroughs, share knowledge, and shape the future of pharmaceutical research and community trials. The program is designed to provide you with the up to date insights into drug discovery and community trials. Renowned experts from academia and industry will share their expertise, unveiling the latest advancements and innovations in the field. The summit is not just about learning, it's also about forging relationships. We believe that engaging in fruitful discussions, meeting potential collaborators, and building relationship will propel your career or business to new heights. Be inspired by our distinguished keynote speakers who will share their wisdom, experiences, and vision for the future of pharmaceutical research and community trials. Dive deeper into the key topics through interactive workshops led by industry leaders, allowing you to gain practical skills and insights. Showcase your research and projects, providing you a platform uh, for constructive feedback and evaluation. We believe that this gathering will serve as a catalyst for innovation and collaboration, ultimately driving advancements that will benefit not only our industry but the global healthcare and communities we serve. We expect that you all enjoy your time in Dubai for the next 2 days, where knowledge exchange, networking goes a long way in defining our pharmaceutical industry and community trials. Let's embark on this transformative journey together. Thank you and have a nice time.
Thank you, sir. Now, I would like to welcome Dr. M. Sridhar R., Vice Chancellor, Chettinad Academy of Research and Education in Chennai, India, to, to deliver welcome address representing Chettinad University.
Greetings from Chettinad Academy of Research and Education. I am Dr. Sridhar, Vice Chancellor, Chettinad Academy of Research and Education. I am delighted to announce that Chettinad School of Pharmaceutical Sciences, Chettinad Academy of Research and Education has entered into an academic partnership with the esteemed Association of Pharmaceutical Research to conduct second Pharma Summit at Dubai, United Arab Emirates, one of the premier international conferences in the field of drug discovery and community trial. This collaboration marks a significant milestone in our ongoing commitment to academic excellence, global engagement and the advancement of knowledge. As an academic partner, Chettinad Academy will play a pivotal role in shaping the discourse, facilitating knowledge exchange and fostering meaningful collaborations within the global academic community. This partnership is a testament to our institution's reputation for excellence and our dedication in contributing to the broader academic landscape. Our faculty and researchers will actively participate in the conference, sharing their expertise and research findings. I look forward to witnessing the positive impact of our participation in the conference. Together, we will continue to strengthen our position as a hub of academic innovation and collaboration. I congratulate the faculty of Chettinad School of Pharmaceutical Sciences for being an academic partner along with the Association of Pharmaceutical Research in successfully conducting this conference. Thank you.
Thank you, sir. Now, I would like to welcome Dr. B. Ramesh, Dean, Academics and Accreditation at Ichunga Chanagari University, India, our academic partner, to deliver a welcome address.
Everyone, my warmest greetings and best wishes. Firstly, as an academic partner, I would like to invite all the delegates and the eminent speakers to this second Pharma Summit to be held in Dubai, UAE on 4th and 5th December 2023. This summit is being organized with the theme Exploring Innovations and Unlocking the Potentials of Drug Discovery. I fervently believe that this summit is the best platform to discuss the research outcomes critically and come up with effective solutions, as well as establish a good collaboration between universities and industrial firms located nationally and internationally to address the current issues. My best wishes for the grand success of this event. Thank you.
Thank you, sir. Next, we'll move on to the next keynote speaker session. Our next keynote speaker of the day, Dr. Nahla Elkurshia Ismail, has taught more than 75 courses to diploma, degree, and postgraduate students in UK and Malaysia using innovative teacher and student-centered learning. As the pioneer of clinical pharmaceutics in Malaysia, she is the founder of the first clinical pharmaceutics research group, CPRG 2008 in Malaysia, and was later acknowledged as Clinical Biopharmaceutics Research Group. Under Research Initiative Group by Research Management Institute, University Technology Mara, UITM, Malaysia. The CBRG comprised of Inhalational Delivery Lab, Biomedical Analysis Lab, and Honey Research Lab. IDL was the first in Asia comprising all pivotal facilities in analyzing particles deliveries emitted from various medical devices, including nasal spray. Metered dose inhalers, phases, dry powder inhalers, and various types of nebulizers, industrial and domestic sprays, as well as environmental air quality assessment ranging from nano to micro sizes of particles. Professor Dr. Nahla holds membership in various local and international pharmacy and health based organizations. She has been publishing more than 300 publications, including research journal articles, proceedings, chapters in books, books and magazines. She has been invited to deliver lectures, talks, keynote, plenary, distinguished speaker, and conduct workshops at local and international events, as well as a reviewer and editorial board member for local and international journals, and serve organizing committee members in various conferences. She is also keenly participating in honey, plant, and animal modeling studies in exploring and defining new therapeutic entity. Professor Dr. Nahla had been awarded research publication and travel grants as well as awards and medals in national and international scientific invention, innovation and design, exhibitions, competitions, conferences, and community services. She is actively promoting health and wellness as well as by being a consultant pharmacist in ambulatory care servicing few private primary healthcare facilities. She is also a freelance senior consultant for medical and pharmaceutical related affairs and entrepreneurships. Now I welcome our keynote speaker of the day, Dr. Naghag Elguch Ismail, Professor and Dean Basmar and Council Member, Malaysian Academy of Pharmacy of Department of Clinical Pharmacy Practice and Pharmaceuticals, AIMST, Malaysia, to deliver a keynote speech. Ma'am, please.
Okay. Good afternoon to all, distinguished guests, ladies and gentlemen. Thank you so much to the chairperson of today, Ms. Nideta, for the kind introduction about myself.
Thank you, ma'am.
Yeah, I'm the Professor Dr. Nahla Afisa Ismail. Currently is the Dean of the Faculty of Pharmacy, AIMS University, Malaysia. So for today, within this 30 minutes allocated to me, I'm going to share with you a little bit on one of the important things that we have to look into when we are going to design an instrument that also known as the questionnaire. I hope all of you can see the slide sharing here. With a short title, Psychometric-Based Instrument Generates Meaningful Outcomes. Things that we're going to cover for today in terms of the review of studies. How are we going to define the construct of the instruments, are also known as the questionnaire, survey, or tool, to ensure that it fits our research of questions or objective. And then the very next important thing to do is the pilot study to test the instruments in terms of the validity and reliability of the instruments. In the early stage of development of any instrument, we have to really look into the conceptualization, okay, of our own study, and this will reflect the way on how we are going to design our instrument. In any study, instrument is created so that we can measure the variables. The unmeasured variables will not be covered under the instrument. Instrumentation is widely used for the quantitative study. Qualitative study also use some kind of instrument, but the outcome of the instrument will not be able to use any statistical analysis compared to the quantitative study. So when we want to design the instrument, number one, what we have to have in mind when we are doing the conceptualization, we have to define what will be the construct. Okay? Construct in other names, also known as the domain. Will there be any domain of our questionnaire, of our instrument, of our survey? Or are they all the items that we are going to incorporate into our instrument are the one that going to define one big construct, one big domain? Or the instrument is actually measuring 2 or 3 or even more domains or constructs? This is very important because it will reflect the variables that we are going to measure. Okay, I mean, if you look at the bottom of the slides, you can see, okay, there will be some conceptualization, okay, that we have to have in mind. What are the things that we want to measure? What will be the domain or the way on how we construct the item? Okay, if like this, we can see like there are actually three main that going to against this one particular construct. Okay, so by saying that, we have to think of number one, whether the construct not directly observable, meaning it's a latent variable. Okay, in this case, we need to develop a multi-item instrument. How about for those items which is observable items, but they are the consequences of the construct? Okay, this is what we call a reflective model. For formative model, we will look into the observable items that are also the determinants of the construct. Okay. Certain construct in an item also look into a multi-dimensional construct. Okay. What does it mean by multi-dimensional construct? Meaning each dimension and its relation to the other dimension should be described. Okay. Another thing that we have to look into while we are developing an instrument, a questionnaire, a survey, a tool, is whether we are adapting or adopting and adopting the instruments that already available in the market. Okay, this is very important for those that thinking, whether if they wanted to create totally a new instrument, or they are creating a newer version of the instrument by adopting another items from another instrument, or they are adapting other items from another instruments. But whatever the method, either is adapting or adopting, or using both adapting and adopting, we have to ensure that the whole item that we develop, it must be able to measure the construct or the domain that we wanted from the study, that which are relevant to answer the question or the objective of the research. There are a few types of measurement that can be involved. Okay. Number one, it can be a self-report. It can be either archival records, or it can be also another type of examinations or observation. Which method to be used, okay, is a choice, whether we want to have a construct form an indissoluble alliance with a measurement instrument. For example, if we want to include an objective measurement to assess the blood pressure, we may use a sphygmomanometer to measure the blood pressure. For physical functioning, it can be measured with a performance test, observation, or with an interview or soft report questionnaire. For performance tests for physical functioning, it is all about the information that obtained about a person can do. And for interview or self-report questionnaire information is obtained about what a person perceives he or she can do. When we are formulating items of an instrument, there are a few things that we have to look into. Number one will be the target population. Why the target population is very important? The way on how the words that been choosing in constructing our item. For example, if the instrument is meant for pediatric, meaning the younger age, or the instrument is meant for adult, or if the instrument meant for elderly, either elderly with disability or elderly without disability. So we have to look into whether the wording can be used for anyone, for the respondent with different type of literacy. So the choice of words and language are very important. Another thing that we have to look into is we have to talk to experts using in-depth interview techniques. Okay? This is very important at the beginning of the developing of the instrument. We need to take some views, some opinions on the items that we have created. At the same time, we have to examine any similar existing instruments. So the skill of the literature review or extracting all different kinds of publications available are very important, because we do not want, we are thinking the instrument is not yet been developed, but actually many instruments have been developed to answer our objective or our question of the particular research. It is okay if you're going to develop an instrument that whereby other type of instrument also available in measuring the same objective of our study. The best part will be we're going to do this validity comparing with the examining similar existing instrument. Another thing that we have to look into is the formulation of the response option. We have to differentiate, for example, if we wanted to measure a knowledge on COVID-19, the type of response is very important. Once it's mentioned measuring knowledge, the option of the response should be either yes, no, or the third one, it can be I don't know. Okay, or true, false, I don't know. And each of those will be given a certain scoring. Okay, why we are giving a certain scoring? Because we wanted to measure knowledge. We wanted to put whether this particular population that we are targeting showed whether they are having good, for example, they're having moderate or poor knowledge on COVID-19. So the type of the response options are very important. If we want to measure like perceived, measure on the awareness, if we want to measure something that we can give some choices, whether people agree or disagree, we can use one of the popular thing will be the Likert scale. Okay, Likert scale either is 3, 5, or 7. Different kind of choices. It can be from strongly agree, strongly disagree, agree, disagree, or it can be also neutral. And for questionnaire, we have to include the instructions to the answer items. If you notice, a very good questionnaire, it will give an introduction about what are actually the instruments that are going to be measured. If there is any jargon words, okay, the words that we cannot simplify, we will put a definition on that particular words so that our respondent, we understand we are measuring this. Okay, and also for certain instrument, we have to think about the appropriate recall period. For example, if we are not doing cross-sectional study, if we are thinking to do longitudinal study, okay, there must be a certain gap between the very first time we are measuring and then in between. For example, 0, it will be the point where we started to give the instrument to measure things that we wanted to measure. Then there will be another gap of 3 months, for example. And then from that 3 months, there will be another gap of 6 months. This is also true if you are thinking to do the reliability study, whereby we have to apply the test-retest. Again, we have to choose what will be the appropriate recall period for the test-retest in order to have a very good test-retest reliability. While we are developing the items, there are a few things we have to avoid. Number one will be the causes of the variable. For example, I just use this example. Attitude toward pharmacognosy. Okay. What does it mean by causes of the variable? It meant that cause of attitude towards pharmacognosy might be how one feels about the course lecturer, but not about the pharmacognosy. For example, the construct of the item is like, I enjoy the pharmacognosy lecturer. Okay. But you wanted actually to measure attitude toward pharmacognosy, not the lecturer. Okay. So this will be considered as a bad item because you are directing the participant to evaluate the lecturer, but not the particular cause of the pharmacognosy. The same goes with the effects of the variable. For example, a student who has a positive attitude is likely to attend class more. We may tempted to write an item like this. I always attend pharmacognosy class. Okay. Why is considered as a bad item? It can be possible for somebody to strongly agree to always attending pharmacognosy, but actually dislike pharmacognosy class. Okay, so this is considered as bad item. Another bad item will be also known as the double barrel items. Okay, we have to make sure that every item focus on only one point at one time. Okay, for example, I give example here. I enjoy pharmacognosy because it is relevant to plantation. Okay, so for this item, there are two things there. I enjoy pharmacognosy and it is relevant to plantation. So what will be people going to select their option? Are they actually like pharmacognosy but not think it is relevant to plantation? So this double barrel item considered as a bad item that should be avoided when we are going to create item in an instrument. In terms of the information of respondents, we have to ensure that the response option must be exhaustive. Okay? Meaning, every participant should be able to find their response in only one and only one category. If we are putting the response as strongly agree, disagree, neutral, disagree, as strongly disagree, it should be the same. It shouldn't be suddenly agree, suddenly the other option will be like not truthful, truthful, no. Response option must be mutually exclusive. Okay? All categories should be mutually exclusive, meaning each of the option is unique from the others. Okay? For response option that must have equal intervals, example using numerical items like age, have to ensure the intervals are equal, okay, unless it is resembled to a certain defined category. For age, usually, we will not put the range of age if we wanted to see what will be the average age of the respondents. For the grouping, we can do the grouping of age later when we are analyzing our findings. Response categories must also be specific. For example, we cannot just simply ask the participant, what is your socioeconomic status? Low, medium, high. What does it mean by low socioeconomic status? What does actually meant by medium or high economic status? It must be specifically designed. In using this example, the number, for example, the monthly income must be stated, the range. Okay? Avoid the social desirability problem. Okay? If, for example, if we want to measure educational level, okay, we cannot put an option there, illiterate or literate. It has to be the specific social desirability. If you're talking about measure educational level, we have to put secondary, primary school, for example, yeah, universities, or if you want to measure the level of the education in terms of the undergraduate or the postgraduate, have to measure the specific. Another thing that we have to think, as I mentioned a little bit in the beginning, is the scoring of the instrument. When we design an instrument, of course, we want to measure something that we can tell to the audience, to the public. We can tell to the readers, what are the things that we are measuring? There must be some definition of the scoring of the instrument. This is very important when we wanted to measure the variable directly, and we're going to directly relate to the construct definition. For example, if we have a multi-item questionnaires that have a 5-point item scales, they're also known as the ordinal scales, are we going to put certain score on that? Often a total score of the instrument is considered to be an interval scale, which makes the instruments more suitable for the statistical analysis. Several things that you have to bear in mind when you wanted to define or to construct the scoring on how we score the items. Number one, can we calculate the scores or the sub-scores? Are all the items actually important, or are we going to use some implicit weight? How are we going to deal later with when there is any missing values? After constructing all those points that we have in mind, before we wanted to test to the actual population that we are targeting, it is highly recommended to run a pilot study. So in this pilot study, we're going to test the instrument that we have developed in a small targeted groups of people. Okay? And this intended to test the comprehensibility, the relevance, and acceptability and feasibility of the measurement instrument. We are going to look into the validity and reliability. There are few methods that I'm going to describe later on how we are going to measure or define the validity and reliability of our study instrument. We always have to bear in mind to allow plenty of time during this pilot study. We have to revise, re-revise, re-revise. Why is so important? Because whatever the outcomes that come from the instrument that we have delivered to our targeted population, number one, it should be able to answer our objective of the study. Number two, it should be able, once we wanted to analyze using any type of statistical analysis, for example, if we're thinking to use the SPSS, we wanted to measure the descriptive and of course the inferential part. And the inferential analysis is the one that we're going to see whether there is any significance between one variable to another. So it's very important to always ensure the instrument, the questionnaire, the survey, the tool that we develop must be meaningful, so that the results that come with it will be considered significantly well presented, well knowledge sharing, and of course, it will be a worth publication in a good impact factor journals. These are some of the psychometric evaluation that we use when we want to measure the validity and reliability. We have the item response theory, the IRT, conventional test theory, CTT. Under the CTT and IRT, both can be used to measure the validity and the reliability. So under the IRT, for reliability, it will measure the items and the response. So for the CTT, it will be measuring the validity and the reliability. Under the validity, of course, under the CTT, we can measure the internal validity and as well as the external validity. For the internal validity, there'll be two types there. Number one is the exploratory factor analysis, EFA, and number two, it will be confirmatory factor analysis, CFA. For the external validity, we can use this convergent and discriminant validity. Let me just use this example of a case study, which is knowledge, attitudes, and practice on uncomplicated malaria management. This project is a part of the PhD project by my former PhD student, now known as Dr. Nanlo Samuel Jimam. I supervised Dr. Nanlo Samuel Jimam when I was in Massa University before I became the Dean for the School of Pharmacy, Faculty of Pharmacy, Ames University. So when we want to develop this instrument, number one, we have to know the background of the study. What are the things actually we wanted to measure? Of course, because we can see there are gaps in terms of the healthcare workers' knowledge, attitude, and practice that can influence their quality of management of the ailments on how they're handling the uncomplicated malaria. Through the extensive review, we found that there was scanty information on valid and reliable instruments that available to assess the healthcare workers' knowledge, attitude, and practice, the KAP, on uncomplicated malaria. So we're thinking to use both classical test theory and items response theory. I'll explain more in detail why we are doing both instead of only one. Many study, they will choose either classical test theory or item response theory. But for our study, we chose both classical test theory and item response theory. To just sum up on all that been written here, okay. Number one, why our study, when we are developing this instrument, we decided to do the reliability and validity using both CTT and RRT. Number one, the CTT, whatever results of the reliability and the validity, same with the result from the IRT, their validity and reliability, we wanted to see whether both are complement each other. Okay, both are testing different things, slightly different things, but the end outcome will be about the same, to see the validity and reliability of the instrument. Number two, because of the sample size that we wanted to test during the pilot study, there are lots of rule of thumb, okay, in the pilot study. All right, I believe that many of you have heard one of the thumb. What is the important point, for the pilot study? It's like one item should be multiplied by 5 participants. So if we have 10 items, 10 multiplied by 5, 50. For example, how about if you have 60 items? Then 60 multiplied by 5, you will get like 300. Whoa. It will be easier if your targeted population is not a population that are limited. For example, if your instrument tested in the general public, there is no detailed inclusion or exclusion criteria, then you may get many respondents for your pilot study using that concept. Okay, one item multiplied by 5. But how about those targeted population? The numbers are not that big. Okay, example, you wanted to test your instrument within a certain targeted vulnerable population. For example, like prisoners, disability people, pediatrics, or adults or geriatric with certain background of diseases. Okay, either the disease are stable or not stable, and many other inclusion and exclusion criteria that comes with it. Of course, the rule of thumb that applying one item multiplied by 5 for each of the item multiplied by 5 will not be feasible, because we use a little big numbers of participants just to make sure that our instrument pass the test of the validity and reliability. This is where we have the IRT. Okay, the IRT, we doesn't have to follow that kind of rule of thumb. Yeah, even a small number of size, okay, between at least between it can be go from 25 up to 50, is already enough to measure the validity and reliability of our instrument. Of course, as I mentioned from the beginning, we have to know what are the things that we measured. Okay, it's very clear. So for this instrument, we wanted to develop and assess the KAP, knowledge, attitude, and practice of the healthcare workers on the uncomplicated malaria, and we imply both psychometric, the classical, and the modern methods. Our research study, we are doing the cross-sectional. So we don't have to think the big gap between the first time we introduced to another, because for this KAP, we're only measuring at that particular time. Okay, And this study location has been conducted in the primary health facilities in Plateau State in Nigeria in 2017. And of course, if we are testing instrument to people, we have to get ethics approval, ethics clearance from the respective authorities. Okay. We have to think also what will be our sampling and sample size calculation. Okay. For our study, since our study is the quantitative in nature, so we have both. We are applying the psychometric method, and for the sampling, because Plateau State is very huge, so we apply stratified random sampling. And of course, after we do the stratification of the sampling, we apply the purposive sampling method for our targeted response that were healthcare workers. These are some of the inclusion criteria, okay, to justify what will be the targeted population, the inclusion and exclusion factors. Okay, all right. From the point of we generated the reliable variables, we prepared the statements, the question of the questionnaire. This is where a draft self-administered instrument consisted of 27 items was developed. Okay, so at the beginning, we have 16 knowledge items, 6 attitude items, 5 practices items, with all the scores, the scoring system. So we define for the knowledge, if they score between 1 to 7, it will consider as poor knowledge, 8 to 11, it will be moderate, 12 to 16, it will be good. The same goes with the attitude. Scoring 1 to 14 will be poor, 15 to 20 will be moderate, 21 to 30 will be good. For practice, between 1 to 12 will be poor practice, 13 to 17 it will be moderate practice, while 18 to 25 it will be good practice. So these are the way on how the scoring, the number of items that we have developed at the beginning of the stage. Okay, we have gone through the phase validity. All right. We also looking into the content validity. The content validity is looking at the relevance, the clarity, the simplicity, and the comprehensiveness of the scale. So for the content validity, we are using the ICVI, which is the Item Content Validity Index, and we're using the SCVI, the Scale Content Validity Index. In terms of the validity, we apply the exploratory factor analysis. Okay, of course, as mentioned for the beginning, the sample size. Okay, you have to look into what will be the sample size that allow the EFA to be used. Okay, looking into the floor ceiling effects and of course the acceptability of the scale. And the eigenvalues greater than 1 rule for the Kaiser criterion have been used. And we are using the factor rotation of the Varimax rotation method. How about the CFA, the complementary factor analysis? We also use this, okay, while using the root mean square error approximation, RMSCA. We also look into the standardized root mean square residual, SMR. And of course, the chi-square minimum discrepancy divided by its degrees of freedom, which is T-mean divided by DF. We also look into the conversion and discriminant validity. In this case, we are using the Fornell-Larker criterion to estimate the conversion and the discriminant validity of the scale. So if the contrast average variance extracted more than 0.5, this will indicate convergent validity. We also looking into the internal consistency and the retest reliability. In this case, we are using the Cronbach's alpha. We set a value 0.60 and above considered as acceptable. And for the retest reliability over time, that was estimated by computing the ICC values, we put 0.79 and above indicated good reliability. For the items response theory, we use the Rush model. This is widely used and been widely cited by Dearborn and Fox, okay, and Lineker. So under the items response theory, we look into the items and persons reliability. We look into the items and persons behaviors consistency by the interpretation of all the following recommended ranges. We have the mean square in-fit and out-fit with the stipulated values. We have the standardized Z values. We have the point measure correlation coefficient value range, item and person relative value, and of course, the separation index value. Okay. We are not going to discuss what are the results of the social demographic characteristic that we have tested among 121 during the pilot study, 121 healthcare workers. The important thing is here, when we're having the scoring for our instrument that measure three main domains, which is the knowledge, attitude, and practice, this is the one. We have a poor, moderate, and good. So you can see here roughly during the pilot study, using 121 healthcare workers, we are looking into the knowledge, attitude, and of course, the practice. Okay. When we look into the content validity, okay, you can see the results here. It shows that high items content validity, ICVI, of more than 0.79. Results also show that high scale CVI between 0.67 to 0.89 and average CVI between 0.94 to 0.98 for relevance, clarity, simplicity, and comprehensiveness of the skill. These are the 27 items of the draft of our instrument before we deleted, after we compared the results using the CTT and the RRT method. Okay. So for the EFA, after we considered the factor analysis in terms of the validity, so from the year 27, it became 18 items. Okay. We deleted the items that considered as the misfit item. So you can see from here, from 27, it became 18 items. Okay. When we're using the goodness of fit, okay, so you can see here the value. Although the chi-square was statistically significant, we always have to remember, because under the CTT, it may be influenced by the sample size. This is the reason why we're also using the IRT when we're analyzing the validity of the instrument. We also tested the convention validity of the 18 item scale, and these are the results that we obtained. When we're doing the reliability, the Cronbach alpha, before and after deletion, okay, we also did the retest reliability on 50 out of those 121 healthcare workers. So you can see here the reliability before and after the deletion. We can see here, for example, the attitudes. After we deleted the 9 items from the 27, it became 18. We tested again the reliability. So you can see here the attitudes from Cronbach alpha 0.59, it became 0.76. The same goes with the practice. From 0.59, it became 0.73. Knowledge from the Cronbach alpha of 0.52, it became 0.61. And you can see here the ICC, the test reliability, it showed better. All are more than 0.9. When we are using the rush analysis, just now showing the 27 items using the factor analysis under the CTT. Now we're using the rush analysis to check the validity. It shows about the similar pattern. before deletion, we have developed the 27 items, and after the deletion, it became 18 items. So both CTT and IRT actually complementing each other. So those misfit items that identified by the rush analysis showed similar about the same item that have been deleted under the CTT of the analysis, validity analysis. Again, using the rush analysis, we can show that the item before and after the deletion, we can see here reliability is much improved in terms of the item and the person reliability before and after the deletion. What we wanted to show here, number one, our instrument that we have developed have been shown very high, reliable, very valid, self-reported outcome measurement that contentedly developed to assess the healthcare workers' knowledge, attitude, and practice on uncomplicated malaria meaningfully, meaning whatever the results that coming out from that, it can be widely accepted, and it is very significant when we wanted to use different type of statistical analysis. That is number one. Number two, when you are creating the instruments, please don't get disheartened by yourself. If let's say you have put many items in, and after you did the analysis in terms of reliability and validity, if those items have to be deleted, by all means, just remove the item. Okay, because our aim to actually develop an instrument that is meaningful, and this instrument, it can be used, whether we are conducting a clinical trials, whether we are measuring certain issues that can be measured. These are the things that we have to take in mind. Just a little bit to thank all of you for your attention. The collaboration between the University of Jos, Nigeria and the AIMS University, Malaysia. And please visit the Malaysian Academy of Pharmacy, whereby this is one of the pharmacy society available in Malaysia. Please visit this portal at the www.acadfarm.org.my. And of course, my university, which is the AIMS University, please do visit our website, and as the team of the university educating tomorrow readers, I would like to thank Association of Pharmaceutical Research, APR, for inviting me to be part of this conference, second Pharma Summit Drug Discovery and Community Trial that conducted hybrid in Dubai. And of course, to the research partner, United Innovators, and the scientific partner, the Chettinan School of Pharmaceutical Sciences and Sri Adichun Charnagiri College of Pharmacy, India. Thank you all, and please be in contact. I'm available at the LinkedIn. Thank you.
Thank you, ma'am. Thank you so much for a wonderful informative session, ma'am. Now I request all the participants to switch on the video for a conference gallery picture, please. Everyone, please. Happy faces all. Thank you so much, everyone. Dear all, I wanted to let you know that we'll take a short break. After the break, we'll continue the sessions. We'll take a short break. After the break, we'll continue the sessions. Thank you, ma'am. Thank you so much. वरती చూడండి Hello everyone. We'll start the session in few minutes.
Dr. Hind with us.
Now, I would like to welcome our third keynote session of the day, Professor Dr. Lakshmi, Professor and Dean, Chettinad School of Pharmaceutical Sciences, Member Board of Management, Chettinad Academy of Research and Education, India. Dr. K. Lakshmi, M.Com, PhD, PGDIPR, is a professor and dean of Chettinad School of Pharmaceutical Sciences, Chettinad Academy of Research and Education, Chennai. She has 23 years of experience in teaching and research. She has received travel grants from AICTE and DST and visited Amsterdam and Turkey for international conferences. She has completed research projects from DST and UGC worth 28 lakhs, 42 lakhs, and 80,000 INR. She has delivered several invited talks in webinars, workshops, and faculty development programs on rights. She has published 74 original research articles in various national and international rights journals. She has presented papers in several conferences and received Best Paper awards. She is also serving as board member and a reviewer in many national and international journals. Her research expertise includes analytical method development and validation using UV visible spectrophotometer, HPLC and HPTLC, chemicometric tools, PLSL toolbox, and OR software. She has completed 11 different MOOCs courses from SWAM and NPTEL. She has authored two book chapters in the book titled Natural Resources for Drug Discovery and one of the authors in the book titled Advanced Pharmaceutical Analysis and published in the year 2020-22. At current institution, she is holding various positions as a member in Board of Management, nominated member NISP Implementation Team, Ministry of Education Innovation Cell, chairperson in Board of Studies, member secretary in Institutional Review Board, and member in Institution Innovation Council. Ma'am, please.
Good afternoon to everyone. Am I audible?
Good afternoon, ma'am. Yes, ma'am, you are audible.
Thank you, Niveda, for a nice introduction.
Thank you, ma'am.
I thank the Organizers Association of Pharmaceutical Research for giving me this wonderful opportunity to connect with all the participants in the second Pharma Summit Drug Discovery and Community Trial. Before COVID, if this same international conference has happened, maybe I would have traveled to Dubai. Thanks to COVID, that we have explored the option of connecting virtually with one another. So actually, there are a lot of differences in technological advancements before COVID and after COVID. Of course, we have lost several lives. So for that, we are not thankful for the COVID. But since this has given us a lot of technological advancements, we should thank for that particular pandemic situation. So being inspired by this technological advancements, I am now going to talk about the current pharmaceutical challenges and the role of AI in that particular aspect. So earlier, our pharma industry was branded as 2.0, and slowly the pharma industry, as well as the pharma education or the education as such, is moving towards education 4.0 or pharma industry 4.0. That is, we are slowly adopting the latest technologies so that we apply the quality by design right from the beginning itself, not checking the quality at the end of the product. So the quality is started. Bringing a quality product, each and every step is started from the scratch. So I am Dr. Lakshmi, Professor and Dean, Chettinad School of Pharmaceutical Sciences, interested in talking about this role of AI in current pharmaceutical industry. So we all know that pharma industry plays a very important role in advancing the healthcare because vaccines have been invented, and this was from India. It was actually manufactured and distributed throughout the world. So with the rapid technological advancement, definitely there are a lot of scope for bringing more innovations in the drug development process. Definitely the manufacturing process is a footprint, and which of course undergoes a lot of modifications in order to suit the current need of the patient. And we have been recently we have been listening to precision medicine. We have been listening to repurposing of the existing drugs. So for all this, the manufacturing unit also have to equip in order to meet this requirements. And many pharmaceutical companies are definitely, they are looking to adopt this newer technologies, including this AI. This in this presentation, I am trying to explore the current challenges in the pharmaceutical landscape and transformative role of artificial intelligence. Of course, in this presentation, I would like to give a disclaimer that all the data that are collected and compiled are from the internet resources, and most of them are from the recent research articles that are published in the year 2023. So, what are the current pharmaceutical challenges the industry faces? As an academician, we know what kind of challenges we face with the 2K kids. So, what are the current pharmaceutical challenges that our industry faces? Of course, the rising drug development cost, the cost, they have to pump in lot of money. In spite of that, the length and the cost of the clinical trials that they have to wait for several years for a product to launch in the market. And definitely there are many complexities that arises for a single drug, to discover a drug. And of course, they have to keep in consideration of regulatory hurdles and the compliances. And what is the major impact of the traditional approaches? As every industry has switched over to the current scenario? No. Of course, the traditional approaches are also being followed, but they face certain limitations, like they could not able to speed up the process. It takes at least 20 years for a drug discovery and development. Every pharmacy students, whenever they join in an institute or whenever they join hands with us, the first thing what they learn is what is drug discovery and development, because we all are striving hard to contribute something in one or the other part of this journey of drug discovery and development. So, inefficiency in research and development can also contribute, can also slow down the drug discovery and approval processes. And it's crucial to recognize the need for innovative solution, because nowadays people, as even the first keynote speaker mentioned, people are using wearable gadgets nowadays. So we are moving forward in most technological advancements. So definitely it is a need to incorporate in our pharma industry also. So don't think this is only in the uh, book level or in the so slowly the pharma industries have started adopting this particular technological advancements. So these are some of this slide actually shows you some of the concepts that are being predominantly used in the pharma industries. So they generally classify this tools. We have lot of AI tools. They generally classify them as supervised learning and unsupervised learning. Like we have classical mode of teaching and online mode of teaching. This AI can be adopted as unsupervised and supervised learning. It's very interesting to learn all this. In supervised learning, it requires a supervisor. That is, the supervisor will frame the pattern and train the AI tool in order to identify the particular, in order to get the output. Say, for example, we have a basket of fruits. So I give a pattern, like if it is round and if it is red in color, then it is apple. And if it is long and yellow in color, it is banana. So I train the system so that that actually adopts that particular concept and it gives the output accordingly. That concept is said to be machine learning and deep learning. And in that, we have different categories: SVM, KNN. SVM is support vector machine, KNN is K nearest neighbor, and random forest is a chart which can predict. So these are all are, of course, supported in various forms. There is no single AI tool which can address all the processes right from drug discovery to development. So according to our need, it can be chosen. So GAN is a generative adversarial network. And all these are neural networks, all GAN, RNN, CNN. Initially, some 5-10 years back, we had a concept called artificial neural network. So that has slowly evolved into RNN, CNN, because here this machine learning and deep learning actually is capable of playing with lot of data, that is big data analytics or data science. So these are the synonyms, these are the names that are very much used nowadays. So unsupervised learning is we need not have to give any directions to the AI tool. So it assesses its own pattern and it gives the information or output related to that particular pattern. So these are some of the examples which is given. Actually, I have an experience working with principal component analysis, PCA, and partial least square. They are called as chemometrics, and they can be effectively used for quantitative estimation of drugs. Simultaneously, we can analyze even 5 to 6 drugs using this particular multivariate analytical tools. So it's definitely a revolutionizing concept in the healthcare, which is offering the new possibilities in the pharmaceutical domain. And this application is not restricted to any one particular uh, series. It applies right from the drug discovery to the clinical trial to regulatory compliance and also to the personalized medicine. That's what we are going to see in the upcoming slides. But everything is just an overview. We are not going into detail of anything. It is just to create some interest in exploring the concepts or learning new concepts from this particular, and we can of course apply all this in our upcoming research. The integration of AI has a potential to address definitely the challenges what they come across. So what is the current trend? It's not from fairy tale to the holy grail. What is happening? So we have been hearing this since 5 years, and recent years we are hearing a lot about this deep learning, machine learning, and the AI. So they are, of course, used in drug discovery and development. These are the 7 areas in which this AI models can be useful, and they can, of course, provide a efficient analytical report. So AI is revolutionizing in the drug discovery process, how it is supporting by virtual screening, molecular modeling, and many of the predictive analysis can be done. precision medicine can also be it can also be applied in that particular domain, and of course for drug repurposing, and even in the formulation, because even any simple person, any simple researcher have already been using I've already been using design of experiment, factorial design for bringing up the drug formulation. But of course, this AI can support in more advanced techniques. Then clinical trial optimization, regulatory compliance and safety, and supply chain optimization. All these are some of the tools in which some of the areas, some of the domain in which AI can support. So, what are all the things that are there in AI in drug discovery? So, it can help in the complex tasks which are happening in the drug discovery. Drug discovery is really challenging, right? So, it can help us, it can support us at different stages of the drug discovery process. So, these are different stages what we have understood. So, we can see here the hit identification, then the target discovery is the first step, then hit identification, then hit to lead optimization, then preclinical studies, and then clinical studies, and then only it gets transformed into a drug. So the AI can accelerate in each and every step. It can help us in target identification and optimizing the screening process, because again, target identification, we need a lot of inputs, the data. So using all this data, it can be easily. And nowadays, we also see that many of the the structure of the drug, the lead optimization, many drug banks are available, many internet resources are available from which the structures can be taken, and using that as an input, we can always work with the available AI tools. So this is some of the examples of successful AI driven showcase the potential of this technology in transforming the industry. I have some of the examples which are in the forthcoming slides. So the application of AI in early stage will also have more efficient and targeted outcomes. You can fix the outcomes at a fixed year or fixed target can be done with this particular AI tool. So these are some of the industries which have partnership with the AI companies, some of the pharmaceutical industries which have the partnership with AI companies, particularly in the process of drug discovery. So here the left column gives you the pharmaceutical organization, various organization, Roche, Sanofi, Bayers, Novartis, AstraZeneca. They have a tie-up with AI because it cannot be done alone. It cannot be done as a standalone industry. Definitely, they need expertise and skills outsourced from other AI delivering organization. So these are some of the companies they actually render their support so that they are using it for drug discovery and clinical trials, and of course bringing out small molecules, finding out new molecules for diabetes and comorbidity conditions, new therapies for rare neurological diseases, and the development of new drugs. These are some of the highlights which I have come across, and it has been taken from a source where they have given the entire information. So these are again other AI tools which can be used in the drug discovery process. Please participants stay muted. So these are some of the AI model tools. This Deep Chem is a open source library. Open source library means it's absolutely free. Anyone can install and then use it, and it gives a wide range of models for drug discovery, including deep learning models. RDKit, it's an open source cheminformatics library, which again gives different functionalities of molecule handling. And the other things are autodoc and then smiles transformer, because in many cases the structure has to be converted into the smiles form and then utilized for docking studies. So in for that reason various tools can be used, and any point of time a single tool cannot solve the purpose. We may have to use multiple tools for a single application also. That is the main challenge that is lying in the AI tool. So AI is also used in the clinical trials. It can optimize the process of patient recruitment by predicting the suitable candidates for clinical trials. So which some one person may be doing manually, and it can also support in predictive analytics, which enhances the understanding of patient outcomes and improving the trial efficiency. So this AI in clinical trials definitely it supports in the cost reduction and faster development timeline. So it actually mainly it minimizes the time which it absolutely minimizes the time. So the traditional approach, this slide actually gives the traditional approach to the clinical development, which is a lengthy process and has a very less success rate. So that's what the comparison has been done in this particular analysis. This is the analysis done by Deloitte, and this analysis is again available in the, sorry, delegates, please stay muted. So for any research and discovery, these are the different phases. It has to come across early phase 1, then phase 2, and then phase 3 and phase 4. So we all have read all these concepts in our drug discovery and development. This is absolutely a traditional approach, but this is not going to, there is no shortcut here. Of course, all these phases will be carried out, but will be carried out in a faster pace. That is the main advantage of using this AI tool. So this is the chart which shows the application of AI enabled technology in the clinical trial. So where the automation is done. So whenever there is some automation is done, definitely it is going to reduce the time span. That is the main purpose of including this AI models in the clinical trials. So advanced data analytics and AI automation can be done in the trial design, so that the accessibility, the feasibility of a patient protocol, everything can be speeded up, so that the performance in the real time monitoring is very good, and the analysis and interpretation can be easily done with a faster time, and then the trial startup, trial conduct, and then the study closeout. So everywhere, every part, the tool is being used. And since the advancement of the mobile application, wearable tools, even the patients can be reminded with the different aspects. The follow-ups can be easier, so that some alerts, some kind of reminders can be given, and then e-tracking can be done using this AI tools. So that is what it says. So that there cannot be any missed out. There may not be the missed out things can be minimized, and even the time can be advanced. So this is the trial enrichment strategies. It tells you how AI technologies can help deliver clinical trial enrichment strategies. This is actually highlighted in the FDA guidance. That's what this particular analysis says. So reduced population heterogeneity. So because the verification is actually done by this AI tool, so that you get some homogeneity in the study what we carry out, so that it increases the study power. and prognostic enrichment, selecting the patient more likely to have measurable endpoints. That is also very important, because we have to select a patient with the measurable endpoints, and there is the patients are identified based on the response to the treatment, and that is again triggered with the help of the apps and other tools. So this ML is the machine learning and deep learning methods can be applied. It can be applied to analyze and interpret the multiple data resources, because we get multiple data sources, and genomics and proteomics data, imaging data. So all this can be together interpreted with the help of this particular source. Here, this picture actually tells about the patient journey through an AI-enabled clinical trial. Because the patient is time to time, they are informed on the progress of the trial in the real time, and because you have a real time application. So AI-enabled algorithms can be utilized, and you adopting this in the digital technologies by using the wearable devices. So these are some of the mobile alerts and wearable devices, and virtual monitoring, telemedicine, by incorporating all these things. So even now, see, the more participants, many participants were preferring to attend the conferences by virtual mode. So this has enabled us many participants, and even nowadays the registrations are, everything is through online. So likewise, the patient enabled platforms can support their cooperation. It can pull out, it can increase the patient cooperation also, and they also feel much comfortable by this AI tools. So next important aspect in which AI tool is used is the dosage form design, because in many of our academic research, we are not much involved in drug discovery, whereas we are involved more in the drug dosage form design. Of course, even the industries have adopted this particular technology nowadays. So it can be used to enhance the nano system, particularly the nano system design. It can expand the present drug testing modeling, and it can also support in the drug repurposing methods. That's what is given. So this chart shows, this picture shows enhanced nano system, how the nano system is enhanced, and selected drug carrier can improve again the ADME concept. Then some specific parameters can be selected for the drug delivery, and sometimes there can be some kind of recognition that can be made for site specific delivery also. These are some of the reported AI and ML tools which are utilized for pharmaceutical product development. So I'm just giving the overview which you can even adopt in your routine research. So genetic algorithms are being used. I think this we have come across since 20 years back. Genetic algorithms, artificial neural network, and there are many research that has been carried out, combination of artificial neural network along with a genetic algorithm. Then artificial intelligence based expert system are nowadays emerging, which adopts the fuzzy logic. So definitely we require a skill support from a person with engineering background, from a person who has a knowledge on all this aspect. And multivariate analysis techniques, this is where I have been working since 10 years. The principal component analysis and partial least square, this actually I am using it for pharmaceutical analysis, but there are many articles in which this are also used for QSAR studies, and it is also used to find out the origin of the plant source, from which geographical origin this particular component is from. Even that can be found out by principal component analysis. So many of you may be working on this, of course. next important aspects where these tools are used is the PBPK modeling. So PBPK modeling itself is a very versatile tool and it has lot of complexities. It is basically done from the inputs that are published already in the general sources, and with that they develop a model, and then a model can uh, again give a output with which we can reduce the uh, animal testing. So in this machine learning can also be given as an input so that it can overcome the bias, and it can give a improvised results. So this is a picture that is taken from a uh, article that I have cited here, a machine learning framework to improve rat clearance predictions and inform physiological based pharmacokinetic modeling, which is published recently in the year 2023. So this is again another article which in which they have used this AI model to predict the delivery, nanoparticle delivery of drug to the tumors in the mice. So this AI and QSAR model is supported. Many were working with PBPK modeling, and again we have a lot of softwares to develop this PBPK model. Along with that, AI and QSAR model can also be employed in order to have a better results and better efficiency. So that's what it says. The prediction can be enhanced. So this AI also addresses the regulatory compliance. It ensures regulatory compliance by streamlining the documentation and reporting processes. So it can also be used in the regulatory compliance also, because quality control and data integrity has to be there, should be linked with this too, and that can in turn enhance the AI application. That can be enhanced by the AI application. So this incorporation of AI in regulatory practices. This is the third session which has a meeting. So the incorporation of this in regulatory practice contributes to the efficient and reliable pharmaceutical landscape. AI can also be used in the personalized medicine. It customizes the treatment plans. Nowadays, you can also see AI tools are being very much utilized by the doctors and for their treatment and personalized medicines. So, tailoring therapies according to the specific patient profiles can be improvised. Because as such, as a by traditional method, it is very difficult for us to read the profiles and predict what kind of modifications are required. So definitely AI can support on this. The era of personalized medicine is enhanced through AI's ability to analyze vast data set. That is the major purpose of adopting the AI tool, because we can work with vast data set, huge number of data. So, of course, though it has lot of applications in different fields, various fields, right from drug discovery to the regulatory compliance, it definitely has some challenges. One has to know the challenges. So, whatever tool we adopt, whichever technique we adopt, whichever technology we adopt, always it is advisable to have a SWOT analysis, the strength, the weakness, the opportunity, and the threats. So here are some of the challenges that are listed. Lack of enabling the data ecosystem, and we have a low intensity of AI research, because the engineering background person can understand the programming, everything, but the connecting tool between the engineering and science is very much needed. Inadequate availability of expertise, technology, and research. This is another major glitch what we have. High resource cost and low awareness for adopting AI in business process. Many times we have a thought to whether to adopt this or not, whether it is cost effective. That is also a big question mark. And definitely there should be some clarity on privacy and privacy of the data which is going to be shared, and because many of the tools are open source tools, So the privacy and security and ethical regulations have to be addressed, and unattractive intellectual property regime to insensitize the research and adoption of AI. These are some of the challenges. But of course, challenges can be overcome by addressing these issues like data privacy and the security concerns. Maybe a person has to ensure this particular aspects, and ethical considerations can be addressed, and ongoing research that ensures the AI implementation so that it can overcome the pharmaceutical challenges. That should be the prime idea behind this. And this is finally, I would like to list the 11 big pharma companies who have partnered with AI. Uh, AI organizations who have partnered with several organizations to implement AI tool in different aspects. So Sanofi has partnered with LE Labs in order to incorporate AI in drug discovery, clinical trials, and manufacturing. These are all the information, authentic information obtained from the internet resource. And Pfizer has tied up, had a partnership with the IBM so that it has launched Paxlovid, an oral drug for COVID-19, which has been uh, found by using this AI tools, because it took only some few months to launch this particular drug. So they have adopted the AI tool, and they say it was launched in 8 months of time. So Novartis has partnered with Microsoft and NIVIDIA, and there are 150 ongoing projects with them. And Jason, the trial 360.ai, and it has more than 100 AI projects. AstraZeneca with OncoShot, and they are working on the systemic lupus erythematosus and the heart failure. And Bayer is assigned with Extensia, small molecule drug discovery, for finding out small molecule drug discovery. And Merck has partnered with Atomwise and Bench Sky for AI drug discovery and development. These are the top companies which have signed, have partnership with AI. And this is my last slide. So definitely there should be some future outlook on this particular tool, because it has a very good potential to revolutionize the pharmaceutical landscape. After 10 years, when we talk about AI, we are going to talk in a different dimension. Whether we like it or not, we have to accept the technological advancements, and this collaborative efforts between pharmaceutical company and AI developers will definitely give a better shape for the future. And there are a lot of ongoing research and developments that continue to uncover the new possibilities for AI in healthcare. So with this, I come to the end of this session, and I thank the organizers for giving me this wonderful opportunity to meet you all by virtual mode. Any questions or?
Participants, if you have any question, you can ask. Or else
മനസ്സിലാകുന്നില്ലേ? നോക്ക്, എന്താ വ്യത്യാസം എന്താ ഇല്ലേ? ഇതിന്റെ അടുത്ത് വരുന്നേ ഇതെല്ലാം ഒരേപോലെ ആവണം, മൂന്നും. അപ്പൊ അതുവരെ നോക്കുക.
Hello, good afternoon, everyone. Can you hear me? Good afternoon, everyone. Good afternoon, everyone. Myself Priyadashni, and I would like to call our today's session chairman. Let's welcome Altana, Professor and Vice Principal, Yapnava Pharmacy and Research Center, India, and Dr. A.L. Akhilandeshwari, Associate Professor, Chettinad School of Pharmaceutical Science, India. And let's start our today's session with the presenter of Gagandeep. Let's welcome Gagandeep Singh from Sardar Bhagavan Singh University to present your paper.
అది ఇప్పుడే కనెక్ట్ చేసా నేను కూడా చూద్దామన్నా. అది ఇంకా 2:30 కదా నాది. సరే,
Mr. Gagandeep Singh, are you there? Hello, sir. Your voice is not audible. Can you please turn on your microphone?
Now I'm audible.
Yes, sir,
Hello.
you're audible.
Okay.
You can.
So drug repurposing, repositioning, reprofiling, or retasking. These are some of the synonyms of the drug repurposing. It is a strategy for finding new uses for the approved or the investigational drugs. It utilizes the drug which are approved by certain regulatory agencies, which are FDA, EMA, MHRA. FDA stands for Food and Drug Administration, the European Medical Agency, and the Medicine and Health Product Regulatory Agencies. These are the agencies by which the drug will be approved. Despite the several advantages, it offers several advantages also. The first one is the risk of failure is very less because the drug is already used. In fact, from the safety point of view, it will get passed. Drug repurposing mainly follows two key concepts. The first one is the same drug interacts with multiple targets, which is also known as polypharmacological targets. The second one is the drug as a target associated disease. These are some significances and the challenges of the drug repurposing. The significance includes the less risk failure, development time gets reduced, and the less investment is needed. The challenges included, which are dose dependency. It is very limited dose dependent. It also has intellectual property issues also, and off-label use of drug is the main drawback of this drug repurposing. This is some comparison between the traditional approaches and by repurposing. When we are using the normal clinical development approach, we will first identify the target drug and fabrication of the molecule. After that, we'll go for the preclinical studies, which includes a very lot of time and money also. After the preclinical studies, it will go for clinical studies and phase 1, phase 2, phase 3. Then after that, the drug will get approved for the clinical use. But when we go for repurposing, if the drug is already approved in the phase 1 or the preclinical trials, we can directly go for the phase 2 trials, phase 2, and after phase 3, the drug will get approved for the clinical uses. These are some of the approaches by which the drug can be repurposed. There are two approaches. There is serendipity, which is by luck or by any means. The second is the hypothesis driven. The hypothesis driven is basically of two types, which are experimentally based or the computer based. The experimentally based drug repurposing is finally done by two methods: binding acid methods or the phenotypic screening, where the computer based can be done by drug centering methods, targeted based method, molecular docking method, or network typing. Experimental approach methods include two type of method, which involve binding assay and the phenotypic assay. The binding assay involve the affinity techniques such as affinity chromatography and mass spectroscopy to find out the drug, that whether it is whether it can be used for the repurpose or not. For example, the acetaminophen is an off target for quinone reductase 2 inhibitor, whereas the phenotypic 2 screening involves the in vitro in vivo disease model, which can be used for the clinical evaluation. This example includes the L-type channel blockers in inhibiting philopodia formation in cancer cells, which states that the calcium channel blockers L-type helps in inhibiting the preventing the cancers. The second is the computational approach. The computational approach can be done by many ways. The first is the drug centering. The drug centering involves the molecule which involve following one of the common theme of the potential target multiple, which involve polyform. For example, if you have one drug and this can be used for multiple targets. This is mainly of drug centric, where the target based approach involves screening a drug molecule with a isolated biological target. This can be explained by, first we have a drug and an isolated target, which can be a protein, a receptor also, anything. In this method, we are checking the affinity for and the binding affinity of the drug with the target. The third is the knowledge base. The knowledge base involves techniques like bioinformatics, chem informatics, and taxonomy. It basically involves the data which are generated from the databases and combining it to form a new method that can be for the drug that can be repurposed or not. This last one includes the network pathway. The network pathway The network pathway involves the it exploits disease omics data, which includes several pathways. It basically involves the drug whose mechanism of action will be studied, and a normal pathway will be drawn, like how in how many ways the drug can be used. These are some of the examples of the repurposed drug which are available. Sildenafil, which is originally indicated for anti-hypertension or angina pectoris, but the new indication is as erectile dysfunction, and the mechanism of action is proposed as phosphodiesterase 5 inhibitor. Itraconazole is an antifungal drug, but is also indicated as is used as anti-cancer also. Nitroxyzine, which is used in UTI infection, it is also has an activity against cancer also. Digoxin, which is used, it is primarily used in congestive heart failure and arrhythmia, but the new indication suggests it has also activity in cancer also. Amphotericin B, which is an antifungal activity but has a new activity in leishmanias also. The tamoxifen is basically used in breast cancer, but the new indication involves antimicrobial, which enhances by mechanism of action enhances the antibody activity of WBCs. The minoxidil is the antihypertensive drug, which is now being used, which can be used for hair growth, regrowth also. By the mechanism, it stimulates follicles movement. The dapsone is for leprosy, and the new indication is malaria. Where sertraline, which is the original indication is depression, but the new indication is antifungal. When we are talking about drug repurposing, we have recently seen some of the very examples and some of the techniques which are recently used in the COVID-19 infection. These are some of the drugs which are repurposed in COVID-19 infection. Sarilumab, which is a primary treatment for rheumatoid arthritis, but it is also repurposed. It has used in COVID-19. With the second is the favipiravir. It is a original indication is influenza, but is also used in COVID-19 also. Third is the remdesivir, which is a broad spectrum antiviral drug, but the new indication suggested its role in COVID-19. The dexamethasone, which is an immunosuppressant, but it also shows its activity in COVID-19 and respiratory distress syndrome. The ivermectin or doxycycline. The ivermectin is a doxycycline. The ivermectin is a parasitic drug, whereas the doxycycline can be used for bacterial infection, which also shows its activity in COVID-19. Whereas hydroxychloroquine. Hydroxychloroquine is an anti-malarial drug, but is also used in COVID-19. This methylprednisolone. Methylprednisolone is also an immunosuppressant, but is also used in COVID-19 also. Whereas a nitroxoxanide is an antiprotozoal drug, which is also used in influenza. Thank you.
Thank you,
Thank
sir.
you.
Our session chairpersons, if you have any questions regarding that, you can ask.
No, ma'am.
Okay. Thank you, Gangandeep Singh, for your presentation. Now let's move on to the next person, Dr. Surend Azad Ramadan, to present from Kumar University of Science and Technology to present his paper.
Okay, thank you for your valuable cooperation. I will try to share my screen. Now, my screen is clear?
Yes, sir, it's visible.
Okay. Okay, before starting, I'd like to welcome all the presenters, and I'd like to send this great achievement to the soul of my mom. Unfortunately, she passed away the previous Monday, and this great participation will be a great achievement for me to be a successful academic person in my academic journey. Actually, in today's presentation, I'm going to talk about one of the most common problem that all the formulation scientists faces from during drug development. Upwards of more than or 40% of all new chemical entities faced pure aqueous solubility, and so many different techniques have been announced to solve this problem that the formulation experts face it from. Actually, using different method will be a great advancement in the term of pharmaceutical drug development. Different techniques can be used to increase or improve the solubility. And one of the novel technique that can be used, and also it's a cost-effective method, which is hydrotropic solubilization technique by using a hydrotropic agent, which alkali metal salt of organic acid. Using a high concentration of hydrotropic agent can lead to increase the or improve the solubility of pure aqueous soluble drug. And also using a mixed hydrotropic agent by using more than one hydrotropic substance will lead to synergetically enhance the or improve the solubility of low aqueous soluble drug. After preparing a different binary, ternary, and quaternary formulation of the atorvastatin calcium as a very slightly soluble drug with different hydrotropic agent, this mixed hydrotropic agent will be used to produce a solid dispersion based on hydrotropic blends. Using different hydrotropic blends to formulate a solid dispersion also will lead to increase the chance of aqueous solubility synergetically. And also, this is the structure of atorvastatin calcium. There is a huge amount of benzene ring and huge amount of the carbon group that will lead to categorize the atorvastatin calcium as a BCS class 2 as a very slightly soluble drug. The main objectives behind this study, this experimental study, was to or were to boost the solubility and dissolution rate on using solvent evaporation method and then physical mixture for forming solid dispersion to increase the chance of aqueous solubility, and then making a comparison or evaluation between the pure sample of atorvastatin with the formulated one. And all the materials that have been used in this study were all in the analytical grades. Before starting to formulating any kind of dosage form, physicochemical characterization or pre-formulation studies are required, including melting point determination, physicochemical identification or organoleptic characterization to determine whether these samples are meet the standardized requirements or not, and then plotting or constructing a calibration curve, then determining a solubility in different methods. And also we had post-formulation studies, including estimation of % of drug content, in vitro drug release or dissolution study, and then to exclude any kind of drug interaction, we had FTIR to exclude any kind of drug interaction, especially or particularly between the atorvastatin and the hydrotropic substance. The novelty of this study was to modify the in vitro dissolution study by using a muslin cloth. Using a powder inside the type 2 apparatus of dissolution study paddle is difficult, and you can't predict the dissolution release or drug release of a substance in the formulation precisely. That's why using a muslin cloth will be a great chance to precisely or properly predict the drug release. Regarding the main findings of this study, if you look at the melting point of atorvastatin calcium and all the hydrotropic agents, actually 5 different hydrotropic agents with different chemical structures have been used, and the chemical structure have a great remarkable impact on improving or on boosting the aqueous solubility of a drug. Resorcinol, sodium salicylate, sodium benzoate, dimannitol, and polyethylene glycol 4,000 degree have been used. And if you look at the melting point reference data with the experimental data, you can realize that there's no any significant shift or no any significant change or discrepancies in the numbers or in the data. This means the standardization or melting point of atorvastatin hydrotropic agent meets the standardized requirement. And also organoleptic properties have been measured or have been determined for atorvastatin calcium and for all hydrotropic agents. First of all, solubility determination or saturated solubility determination of atorvastatin calcium in different media, in different solvents, have been determined to determine whether increasing pH, whether using a salt, whether using other techniques can have a remarkable impact on solubility or increasing solubility of atorvastatin or not. If you look at the saturated solubility of atorvastatin in different solvents and pH-dependent solution, you can realize that there's some change in the solubility. And also we
yes ma'am,
start
கிட்ட கேட்டேன் ஹால் பீன்றது இதுக்கு செப்பரேட் லிங்க் இருக்கா மேம் அப்படின்னு சொல்லி கேட்டேன் மேம். மேம் வந்துட்டு
we started with the binary formulation by using 10% and 20% of different hydrotropic agents. And if you look at the FB6 to FB8, there's a remarkable change in the solubility enhancement ratio or in defaults of solubility enhancement of atorvastatin calcium. And then ternary formulation by using atorvastatin calcium with two different hydrotropic agents have been used, and then this will lead to enhance the solubility enhancement ratio by many folds. And then quaternary formulation by using atorvastatin calcium with three different hydrotropic agent in different ratios from 1 to 3 or 1 to 1 to 3, and then different ratios have been used to determine or to predict whether using a mixed hydrotropic agent can have an extraordinary impact on increasing solubility or not.
Sorry
And
for
also,
the interruption, Dr. Student Azad Ramadan.
Yeah, okay, thank you. My time is finished or not? Dear?
Sorry for the interruption. Yeah, thank you. Thank you for your present. Yes, sir, it's over. Our session chairs. If you have any questions to him, you can ask him.
I'm looking forward to your response and to your question.
Yes, Dr. Azad, what is the rationale for selecting this particular drug?
It's just as a trial, using a different hydrotropic agent with different chemical structure to determine whether it can have a remarkable impact on solubility enhancement or not. Since
Is there any
resorcinol
specific
and other have
reason for
different
selecting atorvastatin? Can it be applied to any other drugs? Can the same method can be applied to any other drugs?
We have different studies that support using hydrotropic solubilization technique, and using, for example, sodium salicylate, sodium benzoate. It was based on the previous studies. The previous studies support using these two hydrotropic agents, and also I have tried to know whether it can have impact on increasing solubility of atorvastatin or not.
Okay, thank you.
Thank you so much.
Okay. Thank you, Dr. Suran Azad for Ramadan, for sharing your valuable insights. Now, let's move
Thanks
on
a
to
lot.
our presenter, Kandapuri Sanjana Gaur from G. Fuller Reddy College of Pharmacy, Hosamani University.
Can I continue now? Can I start? Hello, am I audible?
Yes, ma'am, you can present your paper, ma'am.
Good afternoon, ma'am. Good afternoon, all the co-presenters present here. Today, I want to take up my topic. Okay. Today, my topic is about extraction
Ma'am,
of
your voice.
bale.
I can't hear your voice.
Ma'am, can now? Ma'am, hello? Hello?
Ma'am, your voice is breaking.
1 second, one.
Can you hear?
Yes, ma'am, I can hear you. Can
Yes,
you
ma'am,
hear me?
it's audible, ma'am. You can continue.
It's audible now? Hello? Ma'am,
Yes,
is it audible? Okay, thank you.
Yes, ma'am, you can continue. You can continue presenting the paper.
Okay. My research article is about extraction of bale fruit gum by 3-square full factorial design and application for solubility enhancement of ambrisatin. One second. Okay. Now, the literature review or the other articles that I have referred from various innumerable articles to emphasize the information about the main outlooks of the experiment basing on the brain food gum, its properties, the solid dispersion technique, and the activity in co-experimenting all the derivatives to make the optimized formulation are to be followed by using these authored articles. My study has 3 stages, which include the extraction and characterization using 3-square factorial design for the extraction of bale fruit gum, and then optimization of the solid dispersions, and then optimization of the tablets. Basically, the ambristatin, which shows significant activity, making the workouts last longer without cardiac arrest. Its pathway or the endothelial pathological activity is by antagonizing the endothelial receptor. Then the pre-formulation studies were performed and examined for the physical and the chemical characteristics of the pharmacological substances. Then the analytical calibration curve using water and simulated fluid has 0.1 normal HCl was measured at 269 and 269.2 nanometers respectively. Absorption maxima determination for 10 microgram per ml standard solution of ambristatin scanned on the double beam spectrometer and against water. The extraction of bale gum was performed by using following technique. Then modification of the bale gum to get maximum extraction of the bale gum was done by heating it at 120 degrees for 2 hours. The assessment of this particular gum was done by swelling index and viscosity to obtain maximum outlook from the carrier. Then full 3-square factorial design was performed for the bale gum extraction by using 2 independent factors, temperature and time, which by using 3 different temperatures, that is 65, 75, 85 degrees, and time at 3 frames, 2 hours, 4 hours, and 6 hours. The responses yield and swelling index are taken. And then preparation and assessment of the solid dispersion was done by preparation or by using physical mixture method, kneading method, and also the solvent evaporation, which is by stirring at room temperature while allowed to be evaporated using ethanol. Then evaluation of the extracted gum was done by using spectral scan of ambristatin in water and 0.1 normal HCl. The swelling index was modified for modified bale gum, and the normal bale gum was done. You can see visible results where the modified bale gum has very less swelling index, whereas the bale gum which was normally taken has maximum swelling index. These are the spectral scans that I have performed and taken by using UV spectroscope for ambristatin in water and ambristatin in 0.1 normal HCl. These are the formulations for physical mixture, kneading mixture, and solvent dispersion using these particular ratios of drug and carrier. Carrier, which is bale gum, and drug, which is ambristatin, are taken and performed. The weight of the drug and weight of the carrier are resultant. And then after performing these, I have performed the solubility content uniformity in vitro drug release, where examined for the solid dispersions. All the 3 types of solid dispersions in each 5 formulations were done, and the drug excipient compatibility studies by using FTIR, EXRD, and SEM were done. And then I saw maximum solubility or maximum solubility index for solid dispersion, solid evaporation dispersion for bale gum 10, which is this formulation. And then the formula 1, formula 2, formula 3, formula 4, and formula 5 for different ratios of the excipients are taken and formulated. Now, the next stage of my experiment included the stage design expert for optimizing bale gum extraction using these variables. The 9 runs, which is a 3-square factorial resultant, were taken, and all the 9 runs were performed with 3 temperatures and 3 time frames, and all the 9 resultant yield and swelling index are taken. And to optimize this for the best fit summary was taken by ANOVA quadratic model for both response 1 and response 2. By using ANOVA significant model reduction is done to optimize the model. Then you can see my results over here for response 1 yield. These are the counterparts where the desirability is maximum at the red or towards the red side visibility, and towards the red side visibility, and very less or the less desirability models were seen towards my green or the blue side visibility, where time is taken on the y-axis and temperature at the x-axis. The yield has shown. Then I have taken the predicted versus actual model in a linearity graph with different plots of both the desirabilities. And this is taken as a 3D surface model, 3D surface model for the yield, where these you can see the linearity over here, which shows maximum desirability at the zero, that is 75 75 degrees temperature, and then there was a significant decrease in the result of yield. Then now let us go to the next model, that is next response, that is swelling index. Swelling index also has the same. There was a very less desirability towards this side of the graph and maximum desirability towards this side of the curve. And then I have taken the same as performed for the yield. I have taken the predicted and the actual models of the swelling index. And then the 3D surface model was shown using the design expert software, which has the linearity of, has the temperature increase, there was a linearity and desirability towards the swelling index of the bale gum. And these are the desirability and other plots that I have taken by using the design expert software. And these are the numerical solutions for time, temperature, time with respect to yield and swelling index. Now let us move towards my next stage of the experiment that I have done. The evaluation of the solid dispersions. This is done by essay and practical percentage yield method. The FTIR, differential scanning calorimetry, powdered X-ray diffraction, surface morphology by scanning electron microscope, dissolution studies, in vitro drug release of the physical mixture, drug studies for the solid dispersion by evaluation solvent evaporation method were performed. And these are performed for each and every formulation that I have done in physical mixture, kneading method, and solvent evaporation technique. These are the various results that I have obtained from the FTIR and differential scanning calorimetry. Separately, I have taken the differential, FTIR for ambristatin, bale gum, and then for the formulation, which is supposed to be the optimum formulation in all the solid dispersions that I have taken. Then same for the differential scanning calorimetry. And these are the PXIRDF results, and then these are the SEM results I have obtained. This is the evaluation for the ambristatin immediate release tablets that I have formulated by using various
Kandukuri, sorry to interrupt.
as such. And then
Sorry
these
to
are the
interrupt, ma'am.
stability studies. And
Hello, sorry to interrupt, ma'am. Please conclude your presentation.
Yeah.
Yeah.
The aqueous extraction has been employed to extract natural hydrophilic bale gum. To get the ambristatin aqueous solubility solid dispersions were formulated utilizing approaches like physical kneading and other methods as shown or has discussed before. And then I found maximum drug and gum in ratio of 1:10 for the solid dispersion, which has release rate about 99.56% in 30 minutes, which was very optimum for the formulation to perform as an endothelial receptor antagonist. And then this was formulated into an immediate release tablet, which is a novel drug using the novel technique. And then the solid dispersion approach to successful increased rate. In the future scope, we can do the future studies need to be performed with optimized formulation to determine pharmacokinetic parameters to be used for the commercial purpose. And then these are the different that I have taken, and thank you.
Thank you, Kandukuri Sanjana Gaur. Thank you for your presentation. Dear session chairs, if you have any question regarding the presentation, you can ask him.
No questions, ma'am.
Good afternoon, ma'am. Thank you.
Okay, thank you, ma'am. Next, we call upon Ishnagiri Pandera from G. Pullareddy College of Pharmacy to give his presentation.
Yes, ma'am. Good afternoon, one and all. My name is I. Fanindra. Today I am going to explain about formulation and evaluation of emulgel of flurbiprofen. In this, the introduction include
Yes, sir. It's not audible.
it's now audible. No?
Yes, no, you can talk now. It's audible.
Yeah. The topical administration is a localized application anywhere in the body through ophthalmic rectal or vaginal route of applications. Generally, most common route is oral route of administration, but it has several drawbacks, which include poor bioavailability due to first-pass metabolism and tendency to produce rapid blood level spikes, leading for a frequent dosing or high level of administration of dose. Gel, it is a water soluble and has high skin permeation rate. Thus, it has excellent anti-inflammatory and analgesic activities comparable to its oral administration. Now, flurbiprofen, it is a propionic acid derivative, which is a non-steroidal anti-inflammatory agent with antipyretic and analgesic activity. The present work is aimed to develop flurbiprofen emulgel formulation, which would attenuate the gastrointestinal related toxicities associated with oral administration. These are the ingredients and the formulation batches that have been used for the preparation of this formulation. And the preparation include methylparaben plus propylparaben and propylene glycol are mixed together, and to this solution drug and ethylene solution is added. This total solution is added to aqueous phase containing 220 and purified water, which is heated up to 80 degrees Celsius, and by slowly stirring this solution of oil phase containing span 20 and light liquid paraffin of 70 degrees Celsius, which is cooled until room temperature. Then carbapol 940 and purified water is added, which is a gel. It is added to this above emulsion by adding glutaraldehyde, mixing continuously, which results in the formation of emul gel. Evaluation. Evaluation tests were performed by pH, spreadability, viscosity, drug content, in vitro diffusion studies, ex vivo permeation studies, drug excipient compatibility studies, and stability studies. These are the FTR graph of pure drug and pure drug with xanthan gum. And dye test is a picture showing below. And in vitro release data of emulgel formulations and ex vivo release data. In vitro release data is performed by France diffusion cell by using phosphate buffer of pH 7.4 and the volume is 20 ml, and the medium covered with water and stir continuously by magnetic stirrer at RPM 600, and the temperature is maintained at 37 plus or minus 0.5 degrees Celsius. And at a predetermined rate, at time intervals, 5 ml of sample is withdrawn and same 5 ml sample is administered into it. And the samples were analyzed spectrophotometrically. Ex vivo release data. In this, animals are taken and the abdominal skin of animals was excised and observed for any wounds or cuts. Fat attaching dermis is removed using scalpel and washed with water. And the skin is stored in minus 20 degrees Celsius for and the skin is used in a week. The skin is kept in the diffusion cell such that the dermis contact with the receptor solution, and 500 ml gel was applied to the stratum corneum, and the receptor compartment maintained by stirring. Then the sample is withdrawn at predetermined time intervals for 24 hours, and the drug content is analyzed by using UV visible spectrophotometer. And results and discussion. In this, the FTR spectra formulations with excipients reveal no interactions between the drug and excipient, and both the drug and excipient peaks were identified and interpret the spectra. Gelling agents in low concentrations give gels with less consistency, whereas high concentrations give good consistency. The formulation shows a size range of 40 micrometers. The pH was found to be in the range of 5.96 to 6.98. All the emulgel preparations indicated good spreadability. The extrudability were good and compatible. Drug content indicates that efficient drug loading and uniform distribution of drug in the formulations. From the in vitro diffusion studies, the formulations of these four FPG 4, FOA1, FOA3, and FOA4 have shown more than 70% of drug release for 8 hours. And conclusion. Emulsions of flurbiprofen were prepared and optimized. All the physiochemical properties of the emulsions were checked and were found to be good. Among all the gelling agents used, xanthan gum based formulation using oleic acid has penetration enhancer give superior drug release compared to other formulations. In vitro release of these FOA4, FOA1, FPG4 and FOA3 have shown more than 75% drug release for 8 hours. In ex vivo studies indicated that the FOA4 formulation with xanthan gum, the concentration of 2% and oleic acid has penetration enhancer has shown better release of flurbuprofil for 8 hours. And these are references that I took from international journals of pharmaceutical sciences and pharmaceutics. And thank you.
Thank you, sir. Thank you for your presentation. Dear session persons, if you have any question regarding the presentation, you can ask to the presenter.
What was the lambda max for this drug for analyzing by using spectrophotometer? What is the lambda max chosen? Can you hear me?
Hello, sir. Isnagiri Pandir, are you there? Your voice is not hearing.
Okay, madam, let us continue.
Okay, thank you. Thank you. Next, let's call upon G. Saravanan Kumar from G. Pullareddy College of Pharmacy to present his paper.
Hello. Good afternoon, ma'am.
Yes, sir. Yes, sir, I can hear you. Sir, please share your screen. Saravanan Kumar, please share your screen, sir.
The previous guy didn't stop this screen sharing.
So now you can present your screen.
Okay. Good afternoon, everyone. My topic is about formulation and evaluation of tizanidine hydrofluoride loaded ethozoles of transdermal delivery. Introduction. The introduction includes transdermal drug delivery system eliminates the gastrointestinal involves in the first pass metabolism of the drug. Tizanidine hydrochloride is a drug that is used as a centrally acting on skeletal muscle relaxant. It is used to treat spasms and tightens muscles. It has a half-life of 2 to 4 hours and a low bioavailability of 40 to 44%. The materials include tizanidine hydrochloride, soy lecithin, phospholipon, absolute ethanol, propylene glycol, methanol, triethanolamine, carbopol, and all other chemical and reagents. Preparation of ethosomes. Ethosomes were prepared by cold method. For preliminary screening of ethosome, different types of phospholipones of different concentration of phospholipids were used. Tizanidine hydrochloride were dissolved in ethanol of different percentages in a covered vessel at room temperature at 700 rpm. Water was heated up to 30 degrees centigrade in a water bath and was slowly added in the lipid mixture and under magnetic stirring in a closed vessel and then allowed to cool to room temperature for 30 minutes. Then probes sonicated to decrease the size to desired levels and stored in a stoppered glass bottle at 4 degrees centigrade. The beaker containing phospholipid and drug in ethanol was closed in with aluminum foil to prevent evaporation of ethanol. This prepared formulation is evaluated for entrapment efficiency and ex vivo permissions for optimization. The evaluation parameters include percentage entrapment efficiency, vesicle size analysis, surface morphology, in vitro drug release studies, ex vivo permission studies, evaluation of ethosomal gel, skin deposition study, skin irritation studies. Huh? These are the graphs showing ex vivo permission study and in vitro drug release. The results includes it was found that increased concentration of soil lecithin increases entrapment efficiency up to 3%. Cyanide and hydrochloride exhibited maximum absorption at 318.0 nanometers. Ethosone prepared with 3% soil lecithin and 40% ethanol in vitro release profile of 72.87% for 24 hours. And ex vivo permeation of 600 micrograms centimeter square for 24 hours. Vesicle size was found to be 83.4 plus or minus 5 nanometers. Zeta potential of the optimized formulation was found to ex vivo permeation. These are the graphs of ex vivo permeation studies of gels and stability studies. The conclusion includes the ethylene hydrochloride loaded ethosomes are formulated and optimized using Saguji design. Results showed that concentration of soil lecithin plays a significant ethosome formulation. Stable unilamellar spherical nano-sized vesicles were obtained. The ethosomes incorporated carbogels indicate that ethosome was stable at 4 degrees centigrade. In conclusion, it was found that ethosomes can efficiently deliver through transdermal route. These are the references.
Thank you, sir, for your sharing your presentation. Dear session chairs, if you have any question regarding to the presentation, you can ask to the presenter.
No,
Okay, thank you. Thank you, sir. Let's move on to the next presenter, Rakesh Chandra Parida from Chettinad School of Pharmaceutical Science. Please present your paper, sir. Rakesh Chandra Parida, are you there, sir? Okay, now let's move on to the next presenter, G. Ganesh from Government Arts College. Please present your paper, sir.
Hello. Hello, ma'am.
Yes, sir. Yes, sir.
Yeah, this is Rakesh.
Yes, sir. Dr. Rakesh Chandra Parida, please present your paper, sir.
Okay. Is it visible, ma'am?
No, sir, your screen is not visible.
Hello.
Sir, your screen is not visible, sir.
Okay. Hello.
Yes, sir. Yes, sir. It's visible now. You can continue the presentation.
Yeah, good afternoon, all of you, and respected the chairperson of this session. Myself, Mr. Rakesh Chandra Parida, Research Scholar, Chettinad School of Pharmaceutical Sciences, Chettinad Academy of Research and Education, Tamil Nadu. Now I am going to represent on my topic dissolution method development and validation of low soluble drug bicultamide from an immediate release tablet. Under the supervision of Dr. T. Devi Ma'am, Professor, Department of Pharmaceutical Chemistry, Chettinad School of Pharmaceutical Sciences, Dr. K. Lakshmi Ma'am, Professor and Dean, Department of Pharmaceutical Chemistry, Chettinad School of Pharmaceutical Sciences, and Dr. Gautam Kumar Jain, Sir, Professor, Department of Pharmaceutics, Royal Institute of Pharmaceutical Sciences, Barampur, Odisha. Now I am going to discuss the main objective of my research work is to select a suitable dissolution method for bicultamide tablet 150 mg, which is the products should be able to get more than the 85% or maximum drug release from the dosage form. Now these are the physical chemical property of the API, the bicultamide. The appearance is the white or almost white fine powder, and molecular weight is for 30 gram per mole. And it's the BCS classification, the class 2 molecules, the low solubility and high permeability drug. And solubility is the practically insoluble in water, freely soluble in acetone, and slightly soluble in anhydrous ethanol and methylene chloride. And it is the non-hygroscopic nature material. And here the characterization of the reference product and test product of the biclotamide tablets. Here the almost the test product which similar to our reference product. Here the description, image, diameter, weight, thickness, hardness and disintegrity are mentioned. The test product which similar to our RLD reference product. Next is the dissolution method of by HPLC, and this is the dissolution method development. The outline that these are the following parameter. The first is the evaluation of saturation solubility data by glutamate, evaluation of seek condition, selection of dissolution method, and comparative dissolution profile of test product and reference product in various pH media. Now next is we're going to discuss the evaluation of saturation solubility data. Here we perform that in different media, and here result is showing that 0.1 normal HCl, 6.8 phosphate buffer and water. There is no solubility in this media, and if we're adding the 0.1 normal HCl in different concent of SLS, then it will be little bit gradually increase, and it will be the showing the more result in the pH 6.8 phosphate buffer plus 3% SLS. It is showing the 0.423, and here the graphical presentation of that different media volume result. Here you can see that 0.423 is the highest solubility in pH 6.8 plus 3% SLS, sodium lauryl sulfate surfactant. From the above data is the practically insoluble in 0.1, 4.5 and 6.8, along with the 1% in different media, and 2% also is the practically insoluble, and is very slightly soluble in 0.1 normal HCl along with the 3% SLS, and pH 4.5 with 3% SLS, and pH 6.8 with 3% SLS. It is showing the very slightly soluble. Then here the evaluation of the sink factor. This is the calculation, the solubility mg per ml into 900 ml is the volume divided by strength 150 mg. And here the showing in different media where sink factor was calculated, and here the 6.8 with the 3% surfactant is showing that more sink factor, means 2.5 times it is showing. That's why the based on this study, the 900 ml of media volume can be suitable for our development of biclotamide tablet 150 mg. Here the selection of the dissolution method was optimized. These are the following parameters: selection of dissolution media, selection of surfactant concentration, selection of apparatus, dissolution medium volume selection, dissolution apparatus, agitation speed. Before we are going to discuss in detail the what are the instrument and what are the chemical and what are the chromatic condition used for the development of biclotamide tablets 150 mg. The instrument, the microbalance, HPLC, dissolution apparatus 2, pH meter, and the chemical like the sodium dihydrogen phosphate for the media preparation, sodium hydroxide for the pH adjustment, sodium lauryl sulfate is the surfactant, the methanol we are using for the diluent, acetonitrile using preparation of the mobile phase. And here the chromatic condition, you are using the ODS 3V 150 mm into 46 m, and the wavelength is 270 minutes, and the run time is 10 minutes. Before you are injecting the sample, first you have to baseline that standard, and then inject the 10 microliter of the diluent as a blank and standard solution, and sample should be the liquid chromatography. And the biclotamide standard and sample, the retention time is the 5.4 minutes. Here the chromatographical presentation is the blank, and here the standard and sample. You can see that this the run time is 10 minutes. Then here that retention time you can reach that 5.4 minute, both standard and sample. Next is the selection of dissolution media. In the experiment were performed on of each 6 tablets using the 900 ml with different medium along with the 3% surfactant were used. And here that the tabular form is you can showing that result in different medium. And here that observe the 6.8, the complete release along with another compared to other media, it is showing the higher release and less percentage RSD. Here you can see the graphical representation, the comparative relation profile. You can see the 6.8, the blue line is showing the more result as compared to the 4.5 and 0.1 normal HCl. That's why the 6.8 finalized the dissolution media for biclotamide. And biclotamide also the Tmax is the more than 9 to 72 hours, and it is the one of the factor for choosing that 6.8 media. Here the selection of surfactant, we are evaluated that optimize the different surfactant, the concentration, and we are evaluated our formulation, the 6.82% and 6.8 2.5% and 6.8 3% SLS. Here also we are getting the more result in 6.83% surfactant as compared to the 2.5 and 2%. If you see the 2%, 2.5, it didn't show the any the complete release, but 6.8 is showing that complete release with less percentage of RSD. So that's why there we selected the 6.8 phosphate buffer with 3% SLS was finalized digestion medium for bicaltamate tablets. The next is the selection of apparatus. The most commonly that apparatus were used for apparatus 1 for pedal and apparatus 2 for apparatus 1 for basket and apparatus 2 for pedal method. And if our formulation is the tablet, is the for the tablet formulation, it is the suitable for the pedal, because if not only the all the tablets I'm telling, the if tablet is the floating or swelling, for that purpose you can use that apparatus 1 basket method. So for that our development we are choosing that pedal suitable apparatus for bicaltamate tablet 150 mg. Here the dissolution medium. Here the different we are already adopted the 900 ml volume, but as per comparison the volume, the 500 ml 900 ml is here the showing the result, but it is the 900 ml showing the more release as compared to 500 ml, because that this is the class 2 molecule, the low solubility drug. The 900 ml is the zinc factor is more, because the 2.538 something that zinc factor showing in 900 ml, and 500 ml is showing the 1.4 zinc factor. So due to the solubility, it is showing the more result in 900 ml as compared to 500 ml. For that, that's why we're choosing the 900 ml for the finalized the dissolution medium. You can see the here the graphical representation that showing the 900 ml is more as compared to the 500 ml. The next is the dissolution of apparatus of agitation speed. Here also we evaluated the 50 rpm and 75 rpm. You can see the result that's showing the 75 rpm is more release as compared to 50 rpm, and also percentage of relative standard deviation also very less as compared to the 50 rpm. That's why we are adapted the 75 rpm for the our development. Next is
Sorry for the interruption, sir. Please conclude your presentation.
One minute, ma'am. These are the comparative dissolution profile of the reference product and test product, and you can see that that our test product is comparable with the reference product, and as the similarity factor is more than the 50. Here is the graphical representation of the different medium test versus reference 6.8, 4.5, and here is the 0.1 normal ratio. And the finally we developed that and adopted that dissolution medium is the for release media 6.8 phosphate buffer plus 3% SLS, apparatus to 75 rpm 900 ml, and time point is 5 minutes to 60 minutes. And the final conclusion, dissolution method of bicaltamate tablet was developed and sample was analyzed by using the HPLC successfully for the treatment of prostate cancer. And these are the references. We followed the different guideline regulatory authority and different review articles. And the main is the EMA guidelines of replacement paper, dissolution testing and acceptance criteria of the FDA, biopharmaceutical classification system, and best behavior from FDA, and the electronic EMA from the UK. Thank you.
Thank you, sir, for your presentation. Our dear session chairs, if you have any questions regarding the presentation, you can ask the presenter.
No questions, madam.
Okay. Thank you, sir. Now next, let's move on to the another presenter, G. Ganeshan from Government Arts College. Ganeshan, please present your paper.
Good afternoon, all. Myself, Ganeshan G, research scholar, under the guidance of Dr. P.S. Sujata Madam, Associate Professor, PG and Research Department of Geology, Government Arts College, Coimbatore. I would like to present my research work on hypercholesteremia effects of Lagostomia speciosa, ethanolic green and red leaf extracts against laric acid and ketogenic diets in female albino rats. Introduction. Hypercholesterolemia is a condition characterized by an excess levels of cholesterol in the blood. Cholesterol is a type of lipid, a waxy molecule that aids in the formation and maintenance of cells, hormones, and certain vitamins. Too much of cholesterol might be harmful. High cholesterol levels make people more vulnerable to heart problems such as heart attacks and stroke. Obesity affects 300 million people globally, with 135 million in India. Objective of the study is to evaluate the anti-hypercholesterolemia potential of Lagostomia speciosa ethanoic leaf extract in obese visitor albino rats induced with ketogenic diet. Figure 1 shows experimental plant Lagostomia speciosa green and red leaves. Qualitative control of experimental plant. The experimental plant L. speciosa was collected from PG Girls Hostel, Government Arts College, Coimbatore. The L. speciosa was identified and authenticated at Botanical Survey of India, Coimbatore. The plant extract preparation method was followed by the Kongadip in 1994. The selected medicinal plant L. speciosa were washed, dried, powdered, and then soaked in ethanol for 3 days. The ethanol extract from leaf samples were used for phytochemical analysis to find out the phytochemical constituents in the plant. Phytochemical analysis. Qualitative phytochemical analysis of the leaves of L. speciosa. Ethanolic extracts were carried out according to the methodology of arboreal trees and events. GCMS analysis was conducted at the South India Textile Research Association, Coimbatore. Table 1 shows phytochemical analysis of Lagostomia speciosa ethanolic leaf extract. The phytochemical analysis, the Lagostomia speciosa ethanolic green leaf extract consists of quinone, which was absent in the Lagostomia ethanolic red leaf extract. The Lagostomia ethanolic red leaf extract consists of saponin, which was absent in the Lagostomia ethanolic green leaf extract. Table 2 shows GCMS analysis of L. speciosa ethanolic leaf extract. Cholesterol is a peak compound present in the ethanolic green leaf extract. 1-butanol 3-methyl acetate is a peak compound present in the Lagostomia speciosa ethanolic red leaf extract. GCMS analysis spectrum. Animal studies. The experimental procedures in these studies were approved by the Institutional Animal Ethics Committee, KMCH College of Pharmacy, Coimbatore. Acute toxicity study. Healthy rats were orally fed with increasing doses of extracts for 14 days. The dose did not produce any toxicity or mortality. Experimental design. The experimental group received a ketogenic diet containing 55 to 60% of fat, 30 to 35% of protein, and 5 to 10% of carbohydrate for 3 weeks. The animals were treated orally daily for 21 days. Group 1 control. Group 2 animals received only ketogenic diet. Group 3 animals received ketogenic diet along with lactic acid. Group 4 and Group 5 animals received ketogenic diet as well as Lagostomia ethanolic green leaf extract low dose and high dose. Group 6 and Group 7 animals received ketogenic diet as well as Lagostomia ethanolic red leaf extract low dose and high dose. Results. Result 1. Analysis of quality control. Phytochemical analysis involves the study of plant-derived compounds to identify various bioactive components. The qualitative and quantitative analysis shows the presence of corosolic acid, lagostomine, tannin, and anthocyanin. Acute toxicity study. Healthy rats were orally fed with increasing doses of extract for 14 days. The dose did not produce any toxicity and mortality. Result for body weight. The initial and final body weights of the control group 159.8 and 189.2. The negative control shows 148 and 324 at the end of the experiment after 42 days. The total percentage of increased body weight is 64% in ketogenic diet compared to control group. Table 4 shows lipid profile of Lagostomia speciosa ethanolic leaf extract. After 21st day, except the control group, total cholesterol, triglycerides, HDL levels are increased. After 42 days, Lagostomia speciosa, ethanolic green and red leaf high doses groups, total cholesterol, triglycerides, and HDL levels are decreased compared to control group. Conclusion. Numerous health benefits of Lagostomia speciosa act its anti-obesity, antioxidant, anti-diabetic, and antibacterial properties have been demonstrated. Many other illnesses, including obesity, are treated with plant sterol. Certain plant-based cholesterol may help reduce blood cholesterol. Our research demonstrates that the therapeutic and ornamental plant L. speciosa ethanol red leaf extract contains active components that may lower the body high lipid content. The phytochemical validation provides the means of creating a medication to treat hypercholesterolemia. Corosolic acid and anthocyanin is a class of flavonoids with anti-hyperlipidemic properties are abundant in the red leaves. We concluded that the impact of giving Lagostomia ethanolic legostomia leaf extracts to rat on a ketogenic diet in order to lower hypercholesterolemia. Thank you.
Thank you, sir, for your presentation. Our dear session chairs, if you have any question regarding the presentation, you can ask to the presenter.
Sir, any reason for choosing female animal rate?
Because I'm choosing because of that obesity and diabetics include induced to UTI. So my work is continue to UTI. That's why I'm choosing female rats.
Okay, sir. Thank you.
Thank you, sir.
Thank you.
Thank you, sir.
Thank you.
Next. Now let's move on to the next presenter, A. Agalya from Government Arts College, Coimbatore. Agalya, please present your paper. Okay, now let's move on to the next presenter, R. Manikandan from Chettinad School of Pharmaceutical Science. Mr. Manikandan, please present your paper.
Yes, madam. My presentation visible?
Yes, sir, it's visible.
Yeah. Good afternoon, everybody. Thanks for this opportunity to present this at this esteemed forum. Myself, a Manikandan presenter. I am a research associate in Chettinad College, and my co-guide is the Dr. Lakshmi Kanagara. And this is the background of my presentation. The topic is that the pharmacokinetic modeling in the dosage form design for the pediatrics. And I would like to cover my presentation on the broad background for this thing. What is the literature review? What is PBPK modeling, FDA perspectives and guideline available, case studies and key takeaway and references. If you look at it, industry is advancing towards the personalized medicine. There is a tremendous efforts over the past decade of off-label use of medicines in pediatrics at an undesirable level of 40 to 90%. And the reason being there is a lot of ethical challenge in conduct of clinical trials in pediatrics. Hence, doctors and physicians make use of the empirical approaches in terms of designing the dosage form for the pediatrics. In that scenario, the application of modeling and simulation has shown a better result to predict the exact dosage regimen to avoid these off-labeling in terms of a dose for perspective. And the challenge for not able to perform a good clinical trial for the pediatric is that because of the restriction use of excipients, taste masking technology, ethical consideration in clinical trials, as well as the low market size of the market volumes and limited regulatory clarity in terms of the conduct of the study. So, and also coupled with the type of dosage form, whatever we have, where we will be able to easily tailor make for the pediatrics. There are certain dosage forms which we may not be able to exactly tailor make to the pediatric age group, such as thin strips, scored tablets, chewable strips, as well as the coated tablets. In all these cases, there is again a violation in the appropriate dosing regimen by allometric scaling. And this is further supported with other aspects that, how you can avoid these non-compliance is that, reducing the dosage of administrations by taking care of delayed or prolonged release dosage forms, and also appropriate dosing in the design at stage itself will enable to overcome all these type of adversities. And there are quite a good examples available in the market like methylphenidate, carvedilol, pancreatic enzymes, carbamazepine, levetiracetam, omeprazole, mesalamine. All these are designed for pediatrics. Nevertheless, not all the dosage forms are designed for pediatrics. So if you look at it, an ethical off-label drug accounts for quite a good percentages in the market. We often claim that off-label use is unavoidable, especially in the children's, and legal justice originate from a ethical justice. Therefore, ethical off-label use can be considered as a legal. We thereby the pediatricians uses the ethical off-labeling of the dosage forms. However, there is a scope to improve this by doing appropriating dosage arrangement by conduct of the clinical trials. But to conduct the clinical trials, we have a challenge in terms of again ethical usage of availability of the pediatric populations. In such a cases, one good option is the pharmacokinetic modeling. Now, if you look out of 399 drugs prescribed to neonates and infants between 1997 to 2010, 28 drugs were only studied as a clinical. So remaining things are being extrapolated or interpolated to meet the dosage for the pediatrics. So there comes what is an alternative, and that alternative, one of the alternative is the PBPK modeling. By means of physiological prediction of the pharmacokinetic modeling, we can, based on the adult population study, we can interpolate for the pediatric dosage form. Thereby, we can do the dosage form design, which would be appropriate for the pediatrics without conduct of the clinical trials. If you look at PBPK modeling, how well the regulators are accepted, it comes out to be that being applied at various stages of the research in case of a new drug development, whereby we could able to account 17% of the pediatrics populations dosage forms are in the recent trend as of October 2014, where the modeling has been applied and the appropriate dosing has been predicted and the same has been approved by FDA. And considering these aspects of multiple drugs coming up through modeling, FDA has come back and issued a guidance in 2018. So thereby, we are now ethically able to design a dosage form by using a PBPK modeling based on the pharmacokinetic data, whatever we have generated in the adult population. And now if I look at it, what is the major steps involved in case of a design of the dosage form for the pediatrics? And first is that especially here I'm going to speak about the modified release dosage forms. We have to determine the pharmacokinetic parameter for the pediatric in adult populations, the pharmacokinetic parameter for the extended release dosage form, and thereby that can be designed and developed by using a in vivo dosing in the adult populations. From there, you develop a model, extrapolate the model, whatever we have developed for into an for pediatric group. Thereafter, validate the model and then arrive at a conclusion that through an IVIVC, whether the dose which we wanted to dose for the pediatric will be appropriate and safe and effective. On this background, a workshop has been conducted by FDA and then paper has been published in 2015, and where we could able to say the interpolation by allometric scaling can go as good, reasonably good, up to 2 years old kids. Below than that, it was not able to be having a substantiating the fact. Hence, it concludes that as effective tools or technology for use of a PBPK modeling for pediatrics. And based on this type of approach, which agency also approved one of the product called carbazepine, where the modeling and simulation has been developed based on the pharmacokinetic data of the adult populations by means of a stoichiometric simulation, including between the subject variability and the covariant effect for monotherapy as well as for the adjuvant therapy. A exact dose has been predicted, and the model-based simulation was performed to apply the target exposure matching to the selected carbazepine dose for pediatric subject to attain the exposure associated with the effective as well as well-tolerated dose in the adults. Based on the adult dose, we could able to arrive at a carbazepine dose selection for children above 4 ages for mono as well as adjuvant therapy without conduct of the clinical trial, and FDA has approved this for effective usage in the market. Similar studies, in order to envisage this approach, we are conducting a research. We have done it at a 2-phase level. First, to understand the PBPK modeling relevances in terms of extrapolation from adult data to the a same adult, how we could predict it based on the physiochemical property. There are a lot of softwares available. We made use of a GastroPlus, the simulation software, and predicted the in vivo profile for aprepitant product from an IV simulation to a peroral. From a peroral dosage form, we could able to understand where is the maximum drug absorbed. So what type of model-dependent formulation development can be made. And the results showed a good agreement between the
Sorry for the
data
interruption,
that is
sir.
observed and the simulated profile.
Please conclude your presentation.
Yeah. And based on this, similarly, we have done an extrapolation for an in vitro as well as an in vitro for an extended release dosage form. And then thereafter brought to the in vivo profile extrapolation based on the data available for a second drug, and where we could find a very good agreement with, as I mentioned, an extended release dosage form of 25 mg and IR dosage form of 25 mg could able to predict in terms of a good correlation with the 2 times IR dosage forms. However, there was certain limitations like hydroxyzine hydrochloride. Available literature was used. The whole simulation was based on the literature reported mean plasma concentration, and PBBK relationship was not available. Through all this, we could able to conclude that by making use of a PBBK modeling, we could bring a good extrapolation and of physiological pharmacokinetic data with the in vivo performance. Thereafter, interpolate for the pediatric populations. The pediatric age group exhibit multiple challenges in terms of administration of the dosage form. Hence, the design of a dosage form becomes more critical, and the conduct of clinical trial is not possible. So there we can make use of this PDA modeling and then extrapolate the data for the pediatric data, a pediatric population, and design a very good dosage form which is safe and effective rather than doing ethical off-labeling. These are some of the literature references we could able to use for this type of predictions, and thank you for the time.
Thank you, sir. Thank you for your presentation. Dear session chairs, if you have
Thank
any
you, sir.
question regarding the presentation, you can ask.
No questions, ma'am. Thank you.
Okay.
Thank you.
Okay, thank you, sir. Thank you, sir. Now, let's move on to our next person, Pamarthi Tejaswi from G. Pulla Reddy College of Pharmacy. Miss, please present your paper.
If my presentation is visible, ma'am?
Yes, ma'am, your screen is visible.
Good afternoon, everyone. My name is Tejasvi from G. Pulla Reddy College of Pharmacy from Department of Pharmaceutical Analysis. Today, the topic of presentation is development and validation of novel FTR method for quantitative estimation of apixaban in bulk and pharmaceutical dosage forms. Let's go into the introduction. The FTR stands for the Fourier transfer infrared spectroscopy technique, which it is an analytical technique measures the absorption of the infrared radiation by a sample. The FTR method is used to identify chemical bonds in a molecular and functional groups in organic and inorganic compounds. FTR is also used for the quantitative analysis by exploiting the relationship between intensity of IR absorption bands and the concentration of the substance. In this research work, we developed a quantitative assessment of apixaban in bulk and pharmaceutical dosage forms. The drug profile of the apixaban, which is approved by the US FDA on 28th December 2012. It is appear in a white to pale yellow in color and freely soluble methanol acetonitrile in organic solvents like DMS4, dimethyl sulfoxide and dimethyl formamide. The melting point is at 238 to 240 degrees Celsius. The mechanism of action of this drug is which inhibits the factor 10A. Then the background of the study is reveals that there are several methods like HPLC, tandem methods like LCMS or MS, and RP-HPLC, RP-UPLC, and UV, but there is no FTR method for estimation of the apixaban. So present work is attempt to develop a new method for quantitative estimation of apixaban by using FTR technique. The main objective of the study is to develop a new FTR method for quantitative estimation of apixaban in bulk and pharmaceutical dosage form, and the developed method is validated according to ICH guidelines. Then the application of the method for analysis of marketed formulation. Then the experimental and the results. In this, the method development involves many steps. For liquid samples, we use a liquid cell, and for solid samples, KBR plus. The FTI spectroscopic method was developed by using FTIR. The steps involved in the method development is first we selection of the spectrum mode. In this, the IR spectrum was taken in absorption mode. Means x-axis we plot a wave number, and y-axis we take absorbance mode. Then the selection of the beam. Here the beam parameter is selected as it is used to switch beam operator. Then selection of apodization, we switch the parameter Hap-Gensler, which gives a good resolution. And selection of the detector, DLA-TGS detector is selected. Selection of the mirror speed, and then selection of the sampling technique, solid pelleting technique is selected. The IR spectrum analysis to functional group assessment. Here the data obtained the FTI spectrum by taking wave number on x-axis and absorption on y-axis. By selecting one functional group frequency, we select a frequency by as the intensity of increases in the concentration, the wave frequency for quantitative analysis of apixaban samples are selected. Apixaban IR spectrum consists of peaks at 3,340 centimeter inverse, 3,310 centimeter inverse, 1680, and 2363 centimeter inverse, which is resembles the C double bond OH test and NH respectively. Among the 3,340 centimeter inverse peak shows clear and intense peak, which increase linearly as the concentration increased, which was selected for the quantitative analysis of apixaban. Then the analytical method validation. Here, according to ICH guidelines Q2R1, the method developed was validated. The parameters involves linearity, accuracy, LOD, LOQ, precision. In the optimized FTR conditions, in measurement mode, we took observance mode. Apodization Hap-Gensler method of making pellets is a pressed pellet technique, and frequency range is 4,000 to 400 centimeter inverse. Peak selection is at 3,483 centimeter inverse. Here the NH peak is highlighted as the structure contains a carboxamide group. So the NH peak is a clear and intense peak. The number of scans is 45. 45 scans. The resolution is 8 centimeter inverse. Then let's go into the analytical method validation parameters. The first one is the linearity. Here the standard calibration data is plotted by performing linear regression analysis. The concentration and the observance, the calibration curve is plotted. The response of the drug found linear in concentration range 20 to 120 microgram per mg. Then we get a data of R2 value and the regression equation. R2 value get 0.9966 and Y regression equation is 0.0048x plus 0.2836. Then the next parameter is LOD and LOQ. These are the sensitivity determining parameters. By taking consideration the standard deviation method, we got LOD. LOD is 5.2 and LOQ 16.4. Then the next parameter is Sandel sensitivity data. For apixaban, this is the lowest concentration we consider. 20 microgram per mg is considered. By using formula Sandel sensitivity, concentration is 0.001 by observation value, we get 0.05. Then the next parameter is accuracy. In accuracy, the accuracy study is carried out by calculating percentage recovery of apixaban standard addition method. Here the percentage recovery was calculated by measuring observance and fitting these values into the regression equation of the calibration curve. Then comes the precision. In precision, it is a report in terms of repeatability and intraday precision. In repeatability, we take 6 replicates of a standard pellets of apixaban, and we plot percentage relative standard deviation where are within the limits. Then the intraday precision, take 3 replicates, 40, 60, and 80 microgram per mg, and the percentage relative standard deviations should be within the limits. then the essay of the dosage form, we have to take a brand name of the drug apixaban Eliquis, have to convert the tablets by triturating and evaporating overnight and collect the supernatant, and have to obtain residue should be made into the pellet, and pellet was scanned in the observance mode. Similarly, the UV also we have to observe by taking working standards in the range of 10 to 30 microgram per ml, and we have to take a blank as a methanol, and we have to take a 279 as a wavelength was selected for further analysis. Then by comparing these two results, comparison of the FT and UV method by student t-test, we analysis whether the validation parameters are in the significance level or not. Then by taking the FT and UV statistical analysis, these two methods at significance level of 5% T calculated less than T critical.
Hello, sorry for the interruption, ma'am. Please give a conclusion.
Okay, the p value probability of rejection should be 0.005. So we concluded that an analytical IR spectrophotometric method was developed is used for the quantitative determination of apexaban bulk and marketed dosage form. It is advantageous over other existing method, and it is simple, inexpensive, and a green analytical technique, as it does not require use of any more solvents. So it is suitable for quality control of both pure and marketed solid dosage form. And similar methods can be developed for the category of drugs for the estimation of formulations. Thank you. These are the references. Thank you.
Thank you for your presentation, ma'am. Our dear session chairs, if you have any question regarding the presentation, you can ask to the presenter.
No question, ma'am.
No questions, ma'am. Thanks.
Okay, thank you.
Priyadarshini, can I ask one question before you proceed further? Say, I am in the hall B. Is it the same link to be used? Because I'm trying to contact Priya, but I'm not able to get it.
Just a moment, ma'am.
Yeah. Because this is hall A going on the presentation. Okay, I'm in hall B. Is it the same thing?
Ma'am, just a moment, ma'am.
Yeah.
Yes, ma'am, it's the same link. You can join.
Yeah, okay.
Okay, let's continue our session. Next, we call upon Kopara Soumya from G. Pulla Reddy College of Pharmacy to present her paper.
Good afternoon. Am I audible?
Ma'am, I can hear your voice.
Okay, ma'am. Thank you. So, good afternoon, everyone. This is Kopara Soumya from G. Pulla Reddy College of Pharmacy, Hyderabad, India. And I'm from Department of Pharmaceutical Analysis. Today, I'm going to present my research work. And the topic of my research work is development and validation of derivative and different spectrophotometric methods for the analysis of ambiscertain pharmaceutical dosage forms. So let's go into the introduction. First, we'll get to know about a spectroscopy, and it is a study of the interaction between the matter and electromagnetic radiations. So UV absorption spectroscopy is the most widely employed technique for quantitative analysis of drug molecules, and it involves a measure of the amount of UV radiation that is absorbed by a substance in the solution. So the main principle behind the absorption spectroscopy is Beer-Lambert's law. And in UV spectroscopy, the instrumentation can have a certain components like source, which is the radiation source, and wavelength selectors, which selects an appropriate wavelength, and sample container, which holds the sample, and detectors, which measures the intensity of radiation. So two spectroscopic methods are used for analyzing ambiscertain in pharmaceutical dosage forms, that are derivative and different spectroscopic methods. So derivative spectroscopy uses a first or higher derivatives of absorbance that with respect to the wavelength for quantitative and qualitative analysis, and it involves conversion of a normal spectrum to a higher derivative spectrum spectrums by differentiating absorbance of a sample that with respect to the wavelength. And coming to the difference spectroscopy, it is applicable to the acidic, basic, or amphoteric drug substances that undergo reproducible spectral changes. And this method involves the measurement of the absorbance differences between two equimolar solutions of the different analyte in a different chemical forms that may exist with the different spectral characteristics. So the main advantages of a derivative spectroscopic method is, the first advantage is that an effective enhancement of resolution, which can be useful to separate overlaying of two or more components in a spectrum. And the second advantage is that a discrimination in favor of the sharpest feature of a spectrum that is used to eliminate interferences by broadband constituents. And the different spectrophotometric methods can be quantitated for comparison with other spectra or analytical data. So coming to the drug profile, the drug we have chosen for this study is ambricertan, that is approved by US FDA in the year 2017 by IP, USP, and BP. So the solubility of this drug is, it is partially insoluble in water, and it is freely soluble in acetonitrile and slightly soluble in DMSO, but it is completely soluble in methanol. The mechanism of action of this drug is, the endothelium binds to the ETA receptor. This prevents binding of endothelin 1 to ETA receptor and causes vasoconstriction. So ambricertan binds to the ETA receptor and inhibits its action and prevents from vasoconstriction. So coming to the literature review, the literature review reveals that HPLC, RPLC, and LCMS in biological fluids and spectrophotometric methods and HPLC tandem mass spectroscopy methods have been reported for estimation of ambricertan in single drug or in combination of dosage forms. So there are no derivative and different spectroscopic methods for this drug. So we have taken this for further work. And the main objective of this study is to develop two new derivative and different spectroscopic methods for estimation of ambricertan in bulk as well as in pharmaceutical dosage forms. And it is validated according to ICH guidelines, ICH Q2R1 guidelines. And coming to experimental and results, first we have to develop a method. So the method development is a process of providing that an analytical method is acceptable for use, which allows simplified procedures to be employed and to verify that they consistently deliver a reliable results. So the main steps involved in this is a literature survey. So literature survey is performed to prevent from duplicate research, so that it may prevent from more usage of resources and waste of time. And the second step of this method is selection of a solvent system. So solvent system has to be selected such that it should not interact with the solute and it should be soluble in the drug. And so here in derivative spectroscopic method, the solubility was checked in different solvents like water, methanol, buffer, and acetonitrile. And here is the overlay of the ambricertan in different solvents. So here we can see that buffer has a maximum absorption in pH 6.8 buffer solution. So it has been selected for method development. Coming to selection of wavelength, a particular wavelength has been selected which have a lambda max highest lambda max. So in the spectrum, we can see that 263 nanometers has a maximum lambda max. And coming to the selection of analytical concentration range and verification of the Beer's law. So the drug solution has shown that a good proportionality between the concentration and absorbance in the concentrations ranges of 20 to 100 microgram per ml. So this range was chosen for verifying the linearity. Coming to the method validation, method validation is a process of providing that an analytical procedure is acceptable for its intended purpose. So the parameters included are linearity, accuracy, precision, LOD, and LOQ. So here is the linearity graphs. First spectrum is for zero order, and second spectrum is for first order derivative. Third spectrum is of second order derivative. Here is a tabular form, includes about concentrations ranging from 20 to 100 micrograms with their respective absorbance. And calibration curve is plotted between absorbance versus concentrations, where the graph was in a linear, and the linearity was checked by preparing calibration standards in the concentration range of 20 to 100 microgram per ml, and it was absorbed absorbance was measured at 263 nanometers, and it can be verified by using linear regression analysis and regression equation. Next, coming to limit of detection and limit of quantification. LOD is nothing but the amount of lowest amount of analyte that is present in a sample that can be detected, and LOQ is the lowest amount of analyte sample which can be quantified.
These are the PubChem IDs. No, you have 1 more minute to go. Okay.
Which can be quantitatively determined with suitable.
After that, we have performed them subjected to Protox ADME screening. Out of this 10, 2 molecules were.
Shall I continue?
Yes, ma'am, you can continue.
So the LOD values of 3.75 microgram per ml and LOQ values of 11.37 microgram per ml. That was determined using standard deviation. And Sandel sensitivity is the concentration of the analyte, which results in absorption of 0.001 in a cell having a path length of 1 centimeters. And here is the value. Next, coming to the precision. Precision is the closeness of agreement between the series of measurements obtained from multiple samples of same homogeneous samples under prescribed conditions. These can be considered at 3 levels, that is repeatability, intermediate precision, and reproducibility. Repeatability is nothing but under the same conditions over a short period of time the results are obtained. And intermediate precision, they are divided into intraday and interday. Intraday is variations of results within the same day that were analyzed. And intraday precision, 3 consecutive days are taken for obtaining the results, and the percentage RSD was calculated for these 3. So the acceptance criteria for this repeatability, intraday and interday precision should not be more than 2. So the result shows that all the calculated percentage relative standard deviation values are below 2. So therefore, the method is repeatable and precise for determination of ambiscertin in pure form as well as in dosage form. Then coming to accuracy, accuracy is the closeness between the true value and the measured value. So it should be assessed using a minimum of 9 determinations over a 3 minimum of concentration levels covering the specific range. So the accuracy for drug substances was taken, and the concentrations were included at 48 microgram per ml, 60 microgram per ml, 72. And here is the percentage RSD values. And accuracy for drug substance was determined at varying concentrations levels in ranging of 80 to 120% by analyzing 3 replicates of each sample. And the percentage recovery is calculated from the drug product where a suitable amount of known standard drug was added at 60 microgram per ml. So the obtained values of recovery studies were within the range of 91 to 107%, which indicates that the proposed method is accurate. And coming to assay of fixed dose combination tablets, we use
Sorry for the interruption, ma'am. Please conclude your paper.
Here is a different spectroscopy method. Here also we do the same thing: selection of solvent, selection of wavelength, and linearity was validated, and accuracy, precision. Here is the same thing. So I want to conclude that the method was validated according to ICH guidelines, and all the peer parameters led to the conclusion that the proposed method is linear, accurate, and precise. And these UV is a more easy method than sophisticated methods like HPLC, and it is easy to handle and it is high sensitivity. So from the result, it can be concluded that the proposed first derivative and difference spectroscopy method is effective for the analysis of ambiscertin in pharmaceutical dosage form and without any interference of other constituents in the formulation. And here are the references that I've taken for this research work. And thank you for giving this opportunity.
Thank you for your presentation, ma'am. Dear session chairs, if you have any questions regarding the presentation, you can ask to the presenter.
No, ma'am.
Okay, now let's move to our next presenter, Saroj Chaudhary, from G. Puller Reddy College of Pharmacy. So please present your paper.
Am I audible?
Am I audible?
Okay. Good afternoon, everybody. This is Saroj Chaudhary from G. Puller Reddy College of Pharmacy. And today I'm here to talk about development and validation of FTIR spectrometric method for the estimation of solifenacin succinate in the bulk and pharmaceutical dosage form. So coming to introduction, infrared spectrum is an important record. It gives the information about the functional groups of a compound. So here there are three regions. We have near IR region, mid IR region, and the far IR region. So the near IR region ranges from 0.8 to 2.5 micron, and the mid IR region ranges from 2.5 to 15 micron, whereas the far IR region ranges from 15 micron to 200 microns. So FTIR stands for Fourier Transform Infrared Spectrophotometer. This is the preferred method of infrared spectroscopy, which measures all the infrared frequencies simultaneously rather than individually as with the dispersive instruments. So the FTIR spectroscopy is preferred over the dispersive method because it is not destructive technique and provides a very precise measurement method, and it can increase the scan speed. Like in every second we can collect a scan, and it's mechanically simple with only one moving part. So coming to the drug profile, here the drug selected is solifenacin succinate, and it is anticholinergic and antispasmodic agent, which has a molecular formula, which has a molecular weight of 480.6 grams per mole, and its molecular formula is C27H32N2O6. It appears as a white or yellow, light yellow color, and soluble in very freely soluble in water, soluble in ethanol, and practically it is soluble in heptane. So this is the structure of solifenacin succinate. And literature review reveals that here the solifenacin succinate is subjected to acid hydrolysis, alkaline hydrolysis, oxidation, and the photodegradation. So this technique is very precise and specific, and the statistical analysis, it proves that this method is suitable for the analysis of solifenacin succinate as bulk drug and in the pharmaceutical formulation without any interference from the excipients and the degradation products. So the main objective of the study is to develop the FTIR method for the estimation of solifenacin succinate, which is present in the bulk, pure and in the pharmaceutical dosage form, and then to validate according to the ICH guidelines and the applications of the developed FTIR method for the analysis of solifenacin succinate tablets. Coming to the experimental and results. So in the method development, depending upon the samples, whether it is liquid or state of the sample, whether it is a liquid or solid, the sampling techniques are used, and the FTIR spectroscopic methods were developed using Fourier transform infrared spectrophotometer. So here are the following steps which are used to develop the method. First is selection of measurement mode. So we know that there are 4 different spectrum modes in FTIR, that is transmittance mode, absorbance mode, power mode, and interferogram mode. So depending upon the purpose, we select the type of measurement mode. Like for the quantitative purpose, we select the absorbance mode, and for the qualitative purpose, we use the transmittance mode. So selection of beam. In the selection of beam, when the switch mirror for the external light of an option is installed, then the beam parameter is used to switch the beam direction. Selection of the detector. So the detector is selected, and then comes to selection of the mirror speed. The speed of the mirror is to be selected. Then the selection of sampling techniques. So in the selection of sampling technique, the sampling depends upon the state of the sample, whether it is gas, liquid, or solid. So in the solid sampling techniques, the mull technique, pressed pellet technique, solid dissolved in the solvent, or the solid film can be selected. Sampling of the liquids can be done by using alkyl halide liquid cells. Next, selection of apodization. So the set of the apodization is used to calculate the power spectrum through the Fourier transform of the interferogram. So the apodization function, it gives the effect on the resolution and the sound to noise ratio of the spectrum. Selection of solvent, and then IR spectrum analysis for the functional group assessment. So solifenacin succinate, it consists IR spectrum consists of the peaks at this ranges, the 2937, 1685, 1425, 1319, 1226, and 1122 centimeter inverse, corresponding to the C double bond O stretching and C double bond C stretching. So among these all spectrums, the 1685 centimeter inverse is considered as the intense peak, which increases the linearity as the concentration increases. Analytical method validation. So the method developed was validated according to the ICH guidelines Q2R1. Here are some of the parameters to be discussed, like linearity, LOD and LOQ, Sandel sensitivity, accuracy, and precision. So the FTIR conditions are the optimized FTIR conditions are set up And coming to linearity, first the working standards were prepared and analyzed in the FTIR spectrum. Then the absorbance of the peak at 1685 centimeter inverse were recorded for these standard solutions, and the standards calibration curves are plotted between the concentration and the absorbance. LOD and LOQ, the sensitivity of the proposed method for measurement of solifenaz and succinate, it was estimated in terms of LOD and LOQ. The limit of detection and the limit of quantification, these are determined using the standard deviation method. And the standard deviation and slope were calculated from the calibration curve, which is established from the linearity. Next, Sandel's sensitivity. The lowest concentration of the solifenaz and succinate is prepared from the standard working standard, and then it is calculated from the formula, that is concentration into 0.001 by absorbance value, and we get the Sandel's sensitivity value. Precision. The precision was established by reporting the repeatability and interday precision. So the repeatability was determined by analyzing 6 replicates of 15 microgram per mg of the solifenaz and succinate concentration respectively, and the percentage relative standard deviation was calculated. Whereas coming to the interday precision of the developed method was determined by analyzing the 3 replicates of different concentrations, and the percentage standard deviation was calculated. FTIR spectrum, this is the FTIR spectrum of the interday precision, the day 1, day 2, and day 3. And coming to the repeatability data, these are the 6 repeatable replicates. Coming to the assay of the dosage form, in assay, the 20 tablets were titurated after taking their average weight, and then the tablet powder, which is equivalent to 1 tablet, was accurately weighed and made up to 100 mg with dried KBR, and then it is titurated well to get the concentration of 100 micrograms per mg. Then further, what we do, we dilute further to prepare the pellets of the desired concentration, and the amount of drug was calculated by the formula, that is SA is equals to concentration into dilution factor into average weight of the tablet by weight of the tablet powder which is taken into the labeled claim of the drug into 100. So here this we get the spectrum of the marketed formulation. Coming to the conclusion, the FTIR spectrum method, it is simple and cost effective, whereas the solvent like small amount of solvent is needed in this, and this method can be used as an alternative method for the HPLC, UV, or the pharmacopoeial methods as a quality control check in the pure and marketed formulations. And here are the references. Thank you.
Thank you for your presentation, ma'am. Our dear session chairs, if you have any question regarding the presentation, you can ask to the presenter.
No question.
Thank you, ma'am. Now let's call upon our next presenter, Seth Ghalim from Tlemcen University, Algeria. Okay, participants, there's an update. Who's not able to
medicinal use. All fermentation of the vegetable juice will be at once checked.
Sorry for the inconvenience. To the participant who are not able to join Hall A and Hall B, please raise a query in the comments. We will assign you manually. Okay. Next, let's call upon Kritika K from SRM Institute of Science and Technology, Katankalattur, Chengalpattu. Madam, please present your screen. Asfakul Islam from Guru Nayak Institute of Pharmaceutical Science and Technology. Please present your paper, sir. Sir, I'm not able to hear your voice. Sir, we can't hear your voice.
Is it audible?
Yes, sir. Yes, sir. It's audible now. You can continue.
Okay. Myself, Asfakul Islam. I am from Guru Nanak Institute of Pharmaceutical Science and Technology, Sodhpur, Kolkata, West Bengal, India. I have today I am going to present my research, which is done in our laboratory. The topic is cytoprotective effect of Nigella sativa L-seed extracts against anti-epileptic drug-induced teratogenicity in Daniel radio model. At first, I am going to take introduction about zebrafish, Nigella sativa oil, and valproic acid. Introduction of zebrafish. Zebrafish are small tropical freshwater fish known for their regenerative abilities, making them valuable in scientific research for studying development and disease. Their transparent embryos are also always for easy observation for internal process. Next, Nigella sativa oil. Nigella sativa oil derived from the seed of Nigella sativa plant is known for the potential health benefit. Next, valproic acid. Valproic acid is an anticonvulsant medication used to treat epilepsy and bipolar disorder. It works by increasing level of neurotransmitter GABA in the brain. The objective of the study. The objective of the study is to evaluate cytoprotective effect of Nigella sativa L-seed extracts on zebrafish embryo against valproic acid, anti-epileptic drug-induced teratogenicity in Daniel radio model. This is the methodology we have used. DPPH assay for NSO extracts. NSO means Nigella sativa oil. The radical scavenging activity of different extracts was determined by using DPPH assay. The decrease in the absorption of DPPH solution after the addition of antioxidant was measured at 517 nanometer, and ascorbic acid used as a reference, which is used in 10 mg per ml DMSO. Drug exposure of zebrafish embryos. This is the process. By this we can insert the drug to the zebrafish embryos. This is the diagram in our laboratory, which is done by under the guidance of our mentor. The process is the test begins promptly after the fertilization with approximately 20 eggs per exposure group placed in distilled water. Using a stereo microscope, fertilized eggs are isolated and moved to the test solution, then transferred to petri dish plates and varying concentration to fulfill the statistical criteria. Next, acute toxicity study. Zebrafish embryos post hour per fertilization, 6 hour per SPF. SPF stand for hour past fertilization, were examined under a microscope and healthy embryos were transferred to the test solution. Control included the DDW and the 1% DMSO. Valproic acid and NSO were tested individually. Combination of valproic acid and NSO are also examined for toxicity teratogenicity effect of over 5 days. Observation include coagulation, lack of somite formation, non-detachment of tailbud, and lack of heartbeat, along with their lethal or sublethal endpoint. This is the diagram of the methodology. And the LD50 study. LD50 study of this is the A valproic acid and B for Nigella sativa oil. Valproic acid had after 24 SPF the value is 58.88 micromole per ml, then 37.15, then 11.22, and then 8.70. It means the value is gradually decreased. For NSO, Nigeria sativa oil, in case of 24 SPF, the value is 1000 microliter per ml, and then after 24 hour HPF, this value is decreased and at 0.871, and then after 72 HPF 0.617, and after 96 HPF 0.468 microliter per mole. So we can observe here the value is gradually decreased. Next, mortality study at different concentration. In case of valproic acid, here the mortality percentage mortality for 6.25 micromole per ml. And then percent in case of this image, it is increase and 12.5 micromole per ml, and then 25, then 50, and 70, then 90 micromole per ml. In case we notice that the value is gradually increased. For in case of NSO, percent mortality for 100 per liter, and then value is increased by the 200, then 300, then 400, and 500, 700, 900, for at last 1000. It we can see there the value is gradually increased, and so we can notice that Nigeria sativa oil is show the cytoprotective effects and against valproic acid. Next, the rate of hatching of zebrafish embryos at different time interval. It is the rate of hatching. And diagram we can put here, and we can see that as 120 SPF, the rate of hatching is very high for the valproic acid. And next, the heartbeat of zebrafish embryos at different time interval. This is the heartbeat. This is the different types of heartbeat in case of different chemical. Next, this is statistical analysis of yolk sac area of zebrafish. It is for 96 SPF, and it is for 120 SPF. Next, embryo length. It is for 96 SPF, the total embryo length of zebrafish, and it is for 120 SPF. Here we can see the no major change in this analysis. And it is for 90. It is for caudal field length at 96 and 96 SPF and 120 SPF. This is for head length. This is for head length. Similarly, this is for head width, and it is for eye diameter. It is for protein estimation. Protein estimation of zebrafish upon administration of different concentration. Protein estimation of zebrafish control versus Nigeria sativa oil versus valproic acid combined treatment, and it is for DPPH assay. DPPH assay for determination of antioxidant activity for Nigeria sativa oil using ascorbic acid as control, and the IC50 value of ascorbic acid is 19.2 microgram per ml, and the IC50 value for NSO is the 217.32 microliter per ml. This is the conclusion. Valproic acid during pregnancy negatively impairs cognitive test scores in children. Zebrafish embryo study shows the valproic acid induced teratogenicity mitigated by the Nigeria sativa oil. Ascorbic acid and the Nigeria sativa oil exhibit antioxidant properties. LD50 study indicates the VPA and NSO toxicity with the prolonged exposure amplifying effect. NSO restores heartbeat, reduce yolk sac, edema, and improves body length, fin development, and head dimension. VPA cause abnormalities reversed by the NSO, eye diameter and eye decrease with VPA partially restored by NSO. VPO degrades protein elevated by NSO. Overall, NSO shows protective effect against VPA-induced toxicity in zebrafish embryos. Next, future aspects. The future aspects is that with the aid of this finding of further biochemical estimation can be done established by NSO has the capacity to prevent DNA degradation from the harmful effect of valproic acid. Pre-incubating the zebrafish embryos with NSO and subsequently administration by valproic acid. It can also be evaluated if NSO has protective effect with the prolonged use or vice versa. Pre-incubating with valproic acid and then the administration with NSO. We can study if the Nigella sativa well has a caliber to alter the prolonged harmful effect of valproic acid. Next, these are the reference. Thank you, ma'am, for this opportunity.
Thank you, sir, for your presentation. Our dear session chairs, if you have any question regarding the presentation, you can ask to the presenter.
No question.
Okay. Okay, thank you, sir. Let's move on. Call upon our next presenter, Kritika K from SRM Institute of Science and Technology, Katankalatur, Chengalpattu. Next, I would like to call upon AP Dr. Krupavaram B from KPJ Healthcare University to present his paper. Sir, your voice is not audible, sir.
Good afternoon, ma'am. Can you hear my voice?
Yes, ma'am. Yes, ma'am. We can hear it.
Can see my screen? Can see my screen, ma'am?
Yes, can.
Okay, thank you. Thank you, ma'am, for confirming. A very good afternoon. So, I'm Dr. Krupavaram from KPJ Healthcare University, Malaysia. And my topic today is on evaluation of Speciea microphylla seed extract on anti-inflammatory activity of an SD rats. Okay, so here the inflammation, as all of us know, inflammation is one of the major and common problem for everyone, and that may be because of many aspects, and one of the main aspect is because of the any bacteria or any activated neutrophils or anything have enter attack by using any means like external or internal inflammation also. Sometimes it can be internal, any inflammatory mediators, or sometimes it can be external also. The inflammation, whatever the source also, usually our inflammation will activate our cytokine production. That cytokine production will leads to inflammation. So in this scenario, we have observed one of the folklore medicine which they're very efficiently using last time since so years they are using this medicine for treating diabetes and hypertension. So in this scenario, we selected this plant which has a lot of phytochemical constituents, valuable constituents are available. So here we aim to evaluate the effectiveness of this Specia microphylla extract, seed extract on anti-inflammatory activity, particularly in the SD rats. And I forgot to change the slide. Sorry, ma'am. Okay, so now our main objective is to find out the effectiveness of the anti-inflammatory, where we try to prepare an ethanolic extract first, then followed by we try to find out some secondary metabolites reconfirmation, whether whatever our expected constituents are available or not, then followed by we do some activity experimental for research part on the to find out this anti-inflammatory activity. After taking this extract, we try to convert into dry, we desiccated by using the heating mantle and other methods. And after this conversion, we divided into 2 doses, which is low dose and high dose, and 200 mg per kg and also 400 mg per kg. And to give inflammation, we selected the paw edema. And how we created edema in the paw, we give carrageenan 0.1 ml of 0.1 ml of 1% carrageenan solution. So we see commonly we give for all the animals, and we divided all the animals into 5 groups. One group we maintain as a normal control, where we don't give any drug or any inflammation, also just maintain with normal saline. Second group we may give negative control, which is a carrageenan only. We don't give any drug here. And the third group we give paracetamol protected or diclofenac. In this scenario, we selected diclofenac sodium, and which is a common standard drug. And also we, as per our extract, also we divided into low dose 200 and 400. So we first initially protected the animal by giving our drugs for different groups, third group, fourth group, and fifth group. And third group also we give diclofenac 1 hour before proceeding. And the inflammation we started checking. So after checking our inflammation settings, we can notice here the inflammation started reducing effectively. And inflammation started reducing effectively. The effect started, if we see diclofenac sodium, it started effect from the, I'll show the result. It will start affecting from the 1.5 hours to 2 hours onwards. But the low dose is not very significant actually. But compared to high dose, it is actually very significant from the 2nd hour itself. From 2nd hour, say 2.5 hours itself, the inflammation started reducing. And from the inflammation, it can show a very high significant reduction until 4th hour also. But when it comes to this low dose, it's not much effective. You can see from based on the, you can see from the picture, we can easily identify the reduction is very obvious, and the results we have tested by using our analytical methods, which is one-way ANOVA. Okay, in our conclusion, what we understand, because it contains a lot of flavonoids, and flavonoid is one of the main reason for to show this neutrophils, all this arachidonic acid controlling or liposynic oxygenase pathway, all these pathways. So what we understand, flavonoid is the main constituent which have this ability to obstruct this eicosanoids pathway, and also it will help to reduce the release of this eicosanoid related neutrophil degranulation. And that once this neutrophil degranulation control already, automatically there will be suppression of inflammatory mediators like prostaglandins and all the lipogenase end products. You can see a lot of cytokine end products we can know, right? All these end products, everything will be inhibited. Finally, our prostaglandins could able to control. This is what our assumption, thinking that this is one of the reason. Another one more important assumption what we have, because we are not done into the each ingredient, we did not study separately. This is a crude extract. This is after preliminary study. Later, by using this preliminary results, we are going to furthering the experiment by identifying which chemical constant is responsible. As per our understanding, flavonoid is one of the main ingredient to control this inflammation. So here we are concluding that our saponins or flavonoids are the main reasons. Usually saponins also may cause this action at the early in the inflammation, but we have our drug has having rich in flavonoids, so we are assuming and thinking that flavonoid could be the reason. However, we really need further studies to reveal what could be the main ingredient, exact ingredient to show this effective against the inflammatory disorders. And that's a conclusion, and thank you.
Madam, have you got any ethical approval?
Yes, sir. We already received before proceeding. We will apply for the ethical approval. After getting ethical approval only, we proceed for the experiment.
Okay, thank you.
Thank you, sir.
Thank you, ma'am, for your presentation. Now, let's call upon our next presenter, Sabha Manvili from Sri Ramachandra Faculty of Pharmacy, Chennai. Sabha Manvili is not available. We'll move on to our next presenter, Raghuvaran Gunda from JNTU Hyderabad. Please present your paper, sir.
Yeah, okay, madam. Just wait a minute. I'm sharing my screen. किधर है? Zoom के बाद. Share screen. Just. Hello. Hello. Am I audible, madam?
Yes, sir, you are audible.
Yeah. So myself, good afternoon. Myself, Raghuvaran, a research scholar, JNTU Hyderabad. So my title is Challenges in the Quantification of Nitrosamine Impurities in the Selected Drugs. So we all are aware about the nitrosamine impurities. So before going to my presentation, I want to go to the history and the introduction and the challenges and method of optimization. So nowadays, we are increasing the tuning of the pharmaceuticals in a nanogram level, the impurities. Next. Next. Okay. Then. So the possible nitrosamine impurities may be sourced from API synthesis and its maybe formulation and the packaging also. So we have to careful with in the different levels. So not only the ROS and we don't need to the packaging material also, because cross contamination with printing inks may happen. That may the traces may be lead to the formation of the nitrosamine impurities. Nowadays, the excipient grades also there are nitrosamine free excipient grades are available. Where can in the certificate of analysis we can obtain that from where it can be synthesized and detailed experimental procedure and manufacturing procedure. So it is clearly explained in the USFDA and EMA guidelines. Also, they have clearly mentioned that the excipients, the route of synthesis also need to be expedited while in the DMF process. So before in the nitrosamine impurities also we have to clearly categorize different types. So where we need to stress and where we don't need to stress. So there is a concept of court of concern, which is highly potent and highly mutagenic in the causing the structural features, which may be the aflatoxin like N-nitroso and alkyl azoxy compounds, and these referred as a court of concern molecules. If we can see any these type of structureness or similarness in our final product or in the any reagents, and if there is any possible formation of these compounds, we need to care utmost care. And the justification for specification of these COC compounds may not be see with the generalized justification. So we need to see the separate justification for the specification. And there is a 1, 2, 3, 4 clauses where we can clearly known mutagen carcinogens, and the second is the known mutagen but unknown carcinogenic potential. And the third one is the alerting structure, but there is a no mutagenicity data. And the fourth one is the alerting structure and same alert and related to the drug substances. And fifth one is the no structural alerts. So we have to categorize the nitrosamine impurities in any one of the following. So accordingly, we need to do the risk-based analysis and risk-benefit analysis for the entire process and the entire excipients and everything. So these are the generally acceptable limits for the nitrosamine impurities. These nitrosamine impurities limits will be taken by if our product contains single nitrosamine impurities or multiple nitrosamine impurities. And it depends upon the duration of the therapy, where how much duration we are going to use that product. So we have to take all this consideration while evaluating the specification for this nitrosamine impurity. This is the ICH M7 approach, where TD50 and everything we need to consider. And these are challenges of nitrosamine analysis in the pharmaceuticals. So in this, the main challenges is the sensitivity and the selectivity and the complex in the light of obtaining the reliable, timely results and reproducibility. High sensitivity must be achieved to fulfill the regulatory requirements, and selectivity also to avoid the interference of the main peak. LCA methods have been developed to cover a wide range of lights, and in particular, GC methods cannot directly detect the N-reso and methyl for aminobutyric acid. And so sample derivation was required, increasing very sample preparation time. It will take many efforts to volatile the samples. And for the determination of the most volatile nitrosamine impurities, headspace sampling technique is preferred. No sample preparation is required. We can directly inject since these are highly volatile nitrosamine impurities. One of the advantages of using the technique is that it removes the complexity of the matrix while improving the selectivity for the compounds of interest and reducing the risk of false positive results. And the major problem in the sample preparation in the nitrosamine impurities is that the sample preparation. So this is particularly very problem with the extended release formulations. Why? Because since the interest of sunlight is very nanogram in level, and the extended release formulation contains any polymorphs and polymers, the solubility will be affected. It is very difficult on it will form the gel formation or lumpiness. So as soon as it is very important to inject the very less sample matrix into the system, so that we can get the less interferences and we can improve the S/N and the reproducibility also. So these are some general peaks. Okay. So the optimization of the method. So we have to take the reference standard, and we have to do the standard and MSMS optimization and chromatography optimization and verification with the calibration curve. So these are general problems. So I have based on this null, I have identified 3 to 4 formulation effects, and I have my work is under the publication. So the drug is then, and it is LC-MS-MS method, wherein I have optimized the oven temperature 40, and these are the runtime, and the mobile phase, the composition is there. So these are the optimized parameters. So it is under review process. So thank you.
Thank you for your presentation, sir. Our dear session chairs, if you have any question regarding the presentation, you can ask to the presenter.
Now,
Sir, what about the validation parameters you have carried out, sir?
So based on it is accuracy and precision and recovery 50, 100, 150%, LVD, LVQ and linearity, madam.
Okay, okay. Thank you.
Yes, thank you.
Thank you, sir, for your presentation.
Thank you, madam.
Now, next, let's call upon our next presenter, Ankit from Dr. P.C. Roy College of Pharmacy and Allied Health Science. So please present your paper.
Is it visible and am I audible?
Yes, sir, you're audible.
Just a second. So, greetings everyone. I will quickly introduce myself. I'm Ankit Chatterjee from Dr. Abhisra College of Pharmacy and Allied Health Sciences. Currently, I'm working as a drug safety associate in IQVIA. Today, I'll like to present my work on 2DQS model of structurally diverse knock-on inhibitors and molecular docking using internal and external compounds. These are the table of contents for this presentation. Firstly, introduction and justification. Then comes the brief introduction about the NUAC1, what is the protein. And after that, methodology, followed by result conclusion and references respectively. To begin with, NUAC1 is a gene that codes for NUAC1 protein, which is involved in various cellular events and induces cancer and fibrosis. So by inhibiting this protein, we could stop the cancerous activity of the cell. In this project, we selected 28 structurally diverse NUAC1 inhibitors, and their IC50 values were noted for the study. After generating, we are dealing with the 2D QSR model, so we only worked with 1D and 2D descriptors here. So after generating 1D and 2D descriptor, QSR model was established using various tools, and model was validated by using 1045 external compounds downloaded from KMBL. We wanted to see if the compounds having good predicted bioactivity also bind with the protein well. So we generated a homology model of MOC1 and talked our potential inhibitors with the protein to see their activities. Now, what is NUAC1? The NU stands for novel and AK indicates the AMPK family, which is adenosine monophosphate activated kinase. NUAC1 is a 661 amino acid long serine threonine protein kinase that acts as a regulator of cessation of cellular division and cellular ploidy by mediating phosphorylation of serine 464 of LAS1. Overactivity of NUAC1 induces overactive leads to uncontrolled cell division. The cell fail to seize the cell division, and that leads to uncontrolled cell division. Thus, inhibiting NUAC1 could lead to cell divide more properly. Thus, it acts as a potential target for cancer. Here comes the methodology. To begin with, 28 NUAC1 inhibitors were selected. Their IC50 values are noted and structure are constructed using ChemDraw software, and their structures were energy minimized using Avogadro software using MMFF94 force field. And 2D QSR model was generated, and the descriptor was first the descriptor was generated using pattern descriptor software, and after that 2D QSR model was generated. And this whole data set was divided into two partitions, which is training set and test set, where the training set comprises of 2/3 of the compounds, and 1045 external compounds downloaded from KMBL was validated using this data set. And dimension reduction techniques like PC and PLS were also performed. On the basis of predicted bioactivity, we selected 12 compounds, 6 from internal data set. Is someone talking? Shall I continue? On the basis of predicted bioactivity, 12 compounds are selected, 6 from internal dataset and 6 from KMBL for molecular docking. Homology model of protein was designed on Swiss model web server using the FASTA sequence we found in Uniport. Uniport ID is given over here. and molecular docking was performed. And the X-ray crystallography structure was not available in PDB back then, so we used this homology modeling thing. And after, just a second. After performing homology model, we run the molecular docking and 2D diagram of interaction images were reported. Pardon me, I'm lagging a little bit. Okay. So come to the results. After eliminating 3 outlier compounds, 16 necessary descriptors were collected to build the model. The multiple linear regression of whole set was performed on Microsoft Excel, and it showed a R-square value of 0.95. The 2D QSR model equation was generated using the coefficient of the whole set of Miller. Generally, coefficient of training set are used for this generating the QSR model, but as we are dealing with a structurally diverse compound and the data set was quite small, so we treated this whole set as a training set. This is the QSR equation, model equation provided over here. Both training set and test set are validated using the model equation. R square PRED was calculated by putting the descriptor values respected data set in model equation, and R square PRED values for training set and test set are respectively 0.7817 and 0.9659. These figures suggest external validation of a external data set created with 1044 compounds from MBL. More than 97% of the NOACON inhibitors were predicted correctly. That is, the predicted bioactivity was greater than 0. You can see the 0 line over here, and the most of the compounds showed a result above 0. Now, PCA and PLS are some dimension reduction techniques. For PCA, the eigenvalue should be greater than 1 for retention of the component. We observe 4 components having eigenvalue greater than 1, and they contribute 92% to our regression model, and even after rotation, they contribute the same thing. You can check these 4 components have eigenvalue greater than 1, and they contribute 92%, and even after rotation, they contribute the same. Like PCA component generated in PLS are also correlated with the descriptors. In PLS, 16 components were obtained and 2 sets of R square are generated. One is R square of Y and another is R square of X. This blue line indicates that R square of Y and red line R square of X. Now coming to homology modeling section, we created a homology model for our protein using the FASTA sequence provided in the UniPort, having a sequence identity of 40.45% and Q-mean value and 93.47% favorable Ramachandran plot with a mole probability score of 1.61. And these binding site data were collected from literature and using deep site web server as well. But these differences were quite negligible, so we used data we found in the literature to dock our protein. We use the grid box in these coordinates. These are the 2D interaction image results of the compounds we dock, and we perform molecular docking using 12 compounds, and out of those 7 compounds show 96 greater than minus 9.6 kilocalorie per mole binding affinity, and these are the compounds, and these are the scores and the name of the compounds over here. From this image, we can assess that active groove lies between chain B and chain A. You can check over here. The groove lies between the dimeric proteins chain A and chain B, and it comprises of amino acid like glutamic acid 182, threonine 211, serine 145, cysteine 213, valine 69, etc. In conclusion, we can say that our model is robust enough to predict activity of NOACON inhibitors. Furthermore, it has observed that molecules having good predicted biological activity showed a good inhibiting activity also with the macromolecule. These are the references literature I used for this project. And thank you for this opportunity.
Thank you, sir, for your presentation. Now, dear session, perchance if you have any question regarding the presentation, you can ask to the presenter.
No question.
Thank you.
Okay, thank you, sir. Now let's call upon our next presenter, Meenakshi from Government Siddha Medical College, Chennai. Ma'am, please present your paper. Meenakshi is having some technical issue. Now let's move to our next presenter, Shalini B from National Institute of Siddha. Shalini is unavailable at the moment. Now let's move to our next presenter, Dr. M. Ramamani, National Institute of Siddha. Please present your paper, sir. Ramani is not available at this moment. Now let's call up our next presenter, Mahalakshmi Muskan from Sardar Bhagwan Singh University. Ma'am, please present your paper.
Okay, ma'am, 1 minute. Good afternoon, everyone. My name is Mahalakshmi Muskan, and I'm from Sardar Bhagwan Singh University. My topic is for the today presentation is synthetic methods for controlling shape and size of metallic nanoparticles. These are my contents. First is introduction, second metallic nanoparticle, advantages, disadvantages, property of metallic nanoparticles, significance of shape and size control, and last is synthetic method. Then coming to the introduction part, what is nanoparticle? Nanoparticle are the small particle typically ranging from 1 to 100 nanometer in size, and it is composed of various materials like metals, polymers, ceramics, or biological substances. Due to their minute size, nanoparticle possess unique and distinct physical, chemical, and biological properties that play a crucial role in various fields, including medicine, electronics, material science, and environmental remediation, due to their special properties and potential application. Then coming to the what is metallic nanoparticle, it is a solid metal particle, also size ranging from 10 to 100 nanometers, when which the particles contain a therapeutic molecule which is dispersed within a polymer carrier, enclosed within a polymer shell, and or connected to the surface of the particle through covalent bonding. Third is the advantages. We have advantages like determine the chemical information on metallic nanoscale substrate, enhanced Rayleigh scattering. Third is the surface enhanced Raman scattering. Fourth is a strong plasma absorption, and fifth is the biological system imaging. We have the disadvantages also here. There is the particle instability, impurity, biologically harmful, explosion, difficulty in synthesis. Then coming to the property of metallic nanoparticle, first is the surface atom. In this, we have first point surface atom percentage and full shell clusters. Full shell cluster structure aligned with magic number of protons or neutrons demonstrate a higher percentage of surface atom due to the arrangement within the atomic nucleus. The second is the effect of cluster size on surface atom. Larger cluster results in a reduced number of surface atoms because central atoms are surrounded by more atoms and those in the inner shell are covered by the atom in the upper shell. Third is the dispersed versus aggregated clusters. Clusters can exist in two states, dispersed or aggregated. Dispersed cluster remains separated while aggregated cluster joined together. Fourth is the surface area and dispersed versus aggregate. Dispersed cluster when equal in quantity to aggregated clusters possess a higher surface area to their separate entities. Fifth is the impact on chemical reactivity. Reactants composed of dispersed cluster demonstrate higher reactivity compared to the aggregated clusters. This increased reactivity stems from the greater surface area, influencing the rate of chemical reaction involving these clusters. Then second is the quantum dots. First, the nanoparticle size and composition. Nanoparticles are misplaced particles in the nanometer range comprising thousands of atoms. These are typically semiconductor material made from silicon and germanium. Second is quantum dot behavior and band theory. Quantum dots are explained through semiconductor band theory. The band gap denotes the energy distinction between the valence bond and conduction band in this material. Third is the semiconductor properties. Semiconductors exhibit a band structure where electrons exit in partially filled band separated by a band gap from the empty conduction band. This characteristic distinguishes them from metals. Fourth is the interaction of molecular orbitals. When dealing with the interacting molecular orbital, the highest occupied molecular orbital known as HOMO of one molecule interacts with the lowest unoccupied molecular orbital of another. These orbitals, known as frontier orbitals, closely aligned in energy, facilitate stronger interaction within molecule. Fifth is the application of quantum mechanics. The energies of mental nanoparticle are described using quantum mechanics with the behavior of HOMO and LUMO orbitals, which represent the outermost boundaries of electron in molecules. This quantum nature governs their energetic behavior. Then we have the significance of controlling the shape and size of metallic nanoparticle. First is the tunable property. Size and shape of the metallic nanoparticle directly influence the physical, chemical, and optical properties. By controlling these parameters, one can tailor these properties to suit specific application. For instance, different shape and size exhibit varied catalytic, electronic, magnetic, and optical behaviors. Second is the enhanced reactivity. Nanoparticle with controlled size and shape possess a higher surface area to volume ratio, leading to increased reactivity. This enhanced reactivity is crucial in applications such as catalysts, where surface interaction are vital for efficient reaction. Third is the targeted application. Here, specific shape and size of metallic nanoparticle advantages in different industry. For example, rod-shaped nanoparticle may be more efficient in certain biomedical applications like the drug delivery due to the ability to navigate through cellular barriers. Then we have optical and electronic properties. Science will influence the optical and electronic property of metallic nanoparticle. For instance, a spherical nanoparticle might exhibit different plasmonics behavior compared to nanorods, leading to variation in the optical response, which is important in sensors, imaging, and photonics. Uniform characteristics ensure consistency and reproducibility in application. Additionally, specific shapes may contribute to the stability of nanoparticle, preventing aggregation and enhancing the shelf life. Sixth is the economic and environmental impact. Precisely controlling the shape and size of metallic nanoparticle can lead to more efficient use of materials and reduced waste, thereby positively impacting cost effectiveness and minimizing environmental concern associated with the production. Then we have the synthetic method for controlling the shape and size of metallic nanoparticle. First, the seed-mediated growth. In this method, tiny seed nanoparticle act as a template for the controlled growth of larger nanoparticle. By adjusting the seed size and growth condition, precise control over the final nanoparticle size and shape can be achieved. Second is the polyol method. In this way, which involves the reduction of metal precursor in a polyfunctional alcohol such as ethylene glycol at elevated temperature. The reaction conditions and additive influence the size and shape of the nanoparticles. Then we have third, chemical reduction. Nanoparticles synthesized by reducing metal salts using chemical agents such as sodium borohydride, hydrazine, or citrate. By manipulating reaction parameters like pH, temperature, and the ratio of reactants, the size and shape of the nanoparticle can be controlled. Fourth is the template-assisted synthesis. This method uses templates or molds to control the shape and the size of nanoparticle. The nanoparticle formed within or around the template, taking its shape. Templates can be made up of various materials such as polymers, protein, or other substances. Then the last is the microemulsion technique. Metallic metallic nanoparticles are formed within a microemulsion system composed of water, oil, and surfactant. This method provided confined environment for nanoparticle growth, allowing control over size and shape. Then we have the 6 electrochemical synthesis. Electrochemical techniques involve the reduction of metal ions at the electrode surface in a controlled electrolyte cell setup. By adjusting the voltage and current density and electrolyte composition, nanoparticle size and morphology can be controlled. Then last is photochemical synthesis, utilizing light or radiation to reduce metal precursor. This method offers control over nanoparticle size and shape by adjusting light intensity, wavelength, and reaction condition. Each of these methods offer unique advantages and allow for precise manipulation of nanoparticle characteristics, enabling tailored properties suitable for specific application across various industries. Thank you.
Thank you, ma'am, for your presentation. Our respected session chairs, if you have any question regarding the presentation, you can ask to the presenter.
What is the difference between nanoparticles and nanocomposites?
Ma'am, nanoparticles have the size ranging from 1 to 100 nanometer, and they are made up of in which we can encapsulate the drug loaded. We can make various metallic nanoparticles, and where the nanocomposite, on the other hand, are composed of distinct components at nanometer scales, and they are mainly they contain a matrix type substances, matrix types, and any other nanostructures can be made.
Okay, thank you.
Thank you, ma'am.
Thank you, ma'am, for your presentation. Now let's call upon our next presenter, Kritika Gupta from Sardar Bhagwan Singh University. Please present your paper, ma'am.
Good afternoon, everyone. My topic is nanoemulsion revolutionizing drug delivery through integration. My name is Kritika Gupta and I am from Sardar Bhagwan Singh University, Balawala, Dehradun, Uttarakhand. My contents are challenges in drug delivery, innovative approach, then aim, introduction, advantages, formulation techniques, applications of nanoemulsion, challenges during formulations, future directions, and conclusions.
Madam, share your screen.
Kritika Gupta, ma'am, your screen is not visible. Please share your screen.
Ma'am is now visible.
Yes, ma'am.
My topic is nanoemulsion revolutionizing drug delivery through integration. My name is Kritika Gupta and I am from Sardar Bhagwan Singh University, Balawala, Dehradun. And these are the contents: challenges in drug delivery, innovative approach, aim, introduction, advantages, formulation techniques, applications of nanoemulsion, challenges during formulations, future directions, and conclusions. Drug delivery faces several challenges that impact the efficacy and efficiency of medications. Here's an overview of some key challenges. Bioavailability. Many drugs have poor solubility or low permeability, hindering their absorption into the bloodstream. This affects the drug's effectiveness in reaching its target site. Next is the targeted delivery. Administering drugs to specific locations in the body while minimizing exposure to healthy tissues remains a challenge. Non-targeted delivery can lead to side effects or reduced efficacy. Third is the degradation and stability. Some drugs degrade rapidly in the body, limiting their effectiveness. Maintaining stability during storage and transport is crucial. Then is the controlled release. Achieving a controlled and sustained release of drugs over time is challenging but crucial for medications requiring a specific dosing schedule or prolonged action. Next is the invasive routes. Invasive delivering methods such as injections or implants can cause discomfort, infections, and patients' non-compliance. Patients' compliance, complex dosing schedules, large pill sizes, or unpleasant taste can affect patients' adherence to medication regimens. Then there is a blood-brain barrier. The blood-brain barrier limits drug delivery to the brain, posing challenges for treating neurological conditions. Then is the biodegradability and toxicity. Materials used in drug delivery system must be biocompatible and non-toxic. Some materials used in drug carriers might have adverse effects on the body. Now, last is the regulatory hurdles. Developing new drug delivery systems requiring meeting stringent regulatory standards, adding complexity and time to the process. Next is the innovative approaches or the aim. Addressing these challenges involves innovative approaches like nanotechnology, target delivery systems, controlled release formulations, and novel drug carrier systems like liposomes or nanoemulsions. These advancements aim to improve drug efficacy, reduce side effects, and enhance patient compliance. Now there is an introduction about the nanoemulsions. Nanoemulsion is a hybrid formulation that combines the properties of nanoemulsions and gels. It's created by integrating a nanoemulsion, which is a stable dispersion of nanoscale droplets of one immiscible liquid within another, into a gel matrix. This integration enhances the properties and functionalities of both systems, resulting in a versatile and efficient drug delivery platform. Then the components and the compositions. First is the nanoemulsion, comprises of tiny droplets of oil dispersed in water, that is oil in water or water dispersed in oil. The nanoemulsion forms the active components of the nanoemulsion. It consists of oily phase, aqueous phase, and emulsifiers. Oily phase like lipids or oils such as triglycerides, essential oils, or synthetic lipids. Then aqueous phase, water or an aqueous solution containing drugs, solubilizers, and other active ingredients. Emulsifiers, surface active agents that stabilize the nanoscale droplets and prevent their coalescence. Then there is gel matrix. The gel matrix serves as the base or a carrier for the nanoemulsion. It's composed of gelling agents, thickeners, and stabilizers. Gelling agents, these are used which solidify the liquid components of the nanoemulsion into a gel. Examples include natural or synthetic polymers like carbomers, cellulose derivatives, or gelanga. Thickeners like added to control viscosity and improve consistency, ensuring proper dispersion and stability of the nanoemulsion within the gel matrix. Then stabilizers are used that are the additional compounds to enhance the stability and structure integrity of the gel, aiding in sustained release and targeted delivery. The integration of the nanoemulsion within the gel matrix provides several advantages. Several advantages including like improved skin penetration, controlled drug release, targeted delivery, increased bioavailability, versatility in drug loading, enhanced adhesion, minimized irritation, cosmetic applications, and enhanced residence time, enhanced stability. Now, the nanoemulsion formulation involves integrating nanoemulsions into gel matrices to create a stable and effective delivery system. Several techniques are employed to develop nanoemulsions, ensuring optimal performance and stability. First is the direct incorporation method. This involves directly mixing performed nanoemulsions into a gel matrix, requires careful adjustment of gel components to maintain nanoemulsion stability. Second is the high energy homogenization, that is which uses the process like utilizes high pressure homogenization or ultrasonication to create nanoemulsions. These nanoemulsions are then combined with a gel matrix to form the nanoemulsion. Third is the in situ emulsification or gelation, the simultaneous preparation of nanoemulsion and gel in a single step. The emulsification process occurs within the gel matrix during gelation, forming the nanoemulsion. Then now the phase inversion temperature method manipulates the temperature to induce phase transitions in the components, leading to the formation of nanoemulsions. The nanoemulsions are subsequently integrated into a gel matrix. Then the chemical cross-linking. which utilizes chemical agents or cross-linkers to link gel components while incorporating the nanoemulsion, enhances the stability and controls the release of drugs from the nanoemulsion. Then there's the microfluidization, utilizes microfluidic techniques to generate nanoemulsions by controlled fluid flow and shear stress. The resulting nanoemulsion is combined with the gel to create the nanoemulsion. Then the surfactant and polymer combination combines specific surfactants and polymers to stabilize nanoemulsions within the gel matrix, optimizes interactions between components to ensure long-term stability. Then these are the applications. Nanoemulsions have diverse applications across various fields due to their versatile nature and efficient drug delivery capabilities. These are the first application is in the pharmaceuticals for topical drug delivery or transdermal delivery or ophthalmic formulations. Then the cosmeceuticals and dermatology, which is used in skincare products or treatment of skin disorders. Third is the nutraceuticals, veterinary medicines, agrochemicals, or research and development. These are the challenges which are faced during the preparations of nanoemulsions, like stability, which is maintaining the stability of nanoemulsions over extended periods is a significant challenge. Preventing phase separation or coalescence of nanoemulsion droplets within the gel matrix is crucial. Then the second is the scalability. The scalability of nanoemulsion production for commercial purpose can be challenging due to various variations in manufacturing techniques and batch-to-batch consistency. Then the third is the optimization of formulation, achieving the ideal balance between gel matrix properties, emulsifiers, and other components to ensure its stability, drug loading, and controlled release profiles without compromising other characteristics. Fourth is the compatibility, ensuring compatibility between gel components and nanoemulsion constituents to avoid interactions that may affect stability or therapeutic efficacy. Fifth is the regulatory compliance, meeting regulatory standards and ensuring safety and efficacy in line with the stringent guidelines set by regulatory authorities. Then these are the future directions, that is the advanced formulation techniques. Continued research aims to develop novel and more efficient preparation techniques for nanoemulsions that enhance stability, reproducibility, and scalability. Second is the tailored drug delivery, customizing nanoemulsions for specific drugs or therapeutic needs, allowing for personalized medicine and targeted treatments. Third is the biocompatible and sustainable ingredients, exploring eco-friendly and biocompatible materials for gel matrices and emulsifiers, focusing on sustainability and reduced environmental impact. Then the smart nanogels, developing smart nanoemulsions with responsive properties such as stimuli, triggered drug release, or site-specific targeting in response to physiological cues.
Sorry for the interruption. Please conclude your presentation.
Okay, ma'am. Then this is the conclusion. The integration of nanoemulsions into gel matrices has emerged as a transformative approach in drug delivery, making a significant advancement in pharmaceutical and cosmetic sciences. Through this integration, nanoemulsions showcase remarkable potential in enhancing stability, improving skin penetration, and providing control and sustained release profiles for a diverse range of drugs. Despite the hurdles, ongoing research, innovative formulation techniques, and advancements in nanotechnology continue to propel nanoemulsions towards greater efficacy, safety, and commercial viability. Further exploration into novel formulation studies, biocompatible materials, and smart delivery will undoubtedly unlock new horizons for nanoemulsions, solidifying their role as a cornerstone in modern drug delivery paradigms. Thank you.
Thank you for your presentation, ma'am. Our respected chair sessions, if you have any questions regarding the presentation, you can ask to the presenter.
No question.
No questions, ma'am.
Okay. Thank you, ma'am, for your presentation.
Thank
you. Now next, let's call upon the next presenter, Ms. Pratna Khaire from Jayoti Vidyapeeth Women's University, Jaipur.
Good evening all. I am Ms. Pahire Pradnya, today presenting the
oral presentation on neuroprotective nanocapsule therapy, a recent advancement in formulation and development. Content, introduction, plan of work, objectives, research methodology, and bibliography. Nanocapsule, this is kind of a nanoparticle, provide unique nanostructure consisting liquid or solid core with a polymeric shell. This is increasing interest in the drug delivery applications. Polymer-based nanocapsule have been widely studied as a potential drug delivery system in the recent year. These are the different types of the nanocapsule. Plan of work, literature survey, then availability of resources that are phytoconstituent, availability of phytoconstituent, excipients, and chemicals for the analysis purpose. Then formulation and development of nanocapsule, characterization and evaluation of the nanocapsule, and then animal activity, this biological evaluation. Formulation of nanocapsule of essential oil and comparison of their neuroprotective activity. Objectives: to obtain nanocapsule of essential oil, to study their functionality during the storage at 4 degree and 25 degrees Celsius, then evaluate functionality about the neuroprotective activity, stability, and changes in particle size, polo dispersivity, and zeta potential. To prepare herbal dosage form that improve the memory and use in treatment of Alzheimer's disease. Method of preparation of nanocapsules. There are different methods. First is the polymerization method, in which the monomers are polymerized in the aqueous solution to form a nanoparticle, followed by the placing drug either by dissolving in medium of polymerization or by the absorption. Then ultracentrifugation method, which has been utilized for the purification of nanoparticle suspension. Then remove the various stability and surfactant employed by the polymerization. Next is the interfacial polymerization, in which this method is alternative to the bulk polymerization. There is a required high temperature is required in this method and comprises the two immiscible solvent, in which monomer in one solvent instantaneously reacting with the monomer to another solvent, and higher molecular weight of monomer are obtained since it is more likely to stumble. The next is the arc discharge method. This is a rarely used method in which the development of modified strategies and new techniques are involved in this. There is a synthesized new type of nanocapsule, which is intermetallic compound are used. For example, aluminum trioxide. This is used for this method, arc discharge method. Then emulsion polymerization. Here the pre-emulsion preparation for one of the nanocapsule provided as a means first of all pre-emulsion preparation is formed, and then pre-emulsion was synthesized by blending the two parts, and then resulting pre-emulsion was cooled and sonicate for the 3000 RPM, and the pre-emulsion that was transferred into the 3 neck round bottom flask, and then mechanical stirrer is used, then reflux condenser. And other preparation method nanocapsule include the electron irradiation deposition method. Then I will perform the characterization of essential oil nanocapsule particle, and the characterization are the particle size and polydispersibility measurement, zeta potential, pH measurement, fluorescence quenching, encapsulation efficiency and loading capacity, stability over time of essential oil loaded nanocapsule, then statistical analysis. Evaluation study, scanning electron microscopy, differential scanning calorimetry, transmission electron microscopy, X-ray diffraction studies, high resolution transmission electron microscopy, X-ray photoelectron spectroscopy, multi-angle laser scattering, FTIR analysis. Microbiological studies, microorganism and growth condition, minimum inhibitory concentration and minimum bactericidal concentration. Then I have selected the L'Oreal leaves to obtain the essential oil. I have taken the powder of L'Oreal leaves and subjected to hydrodistillation by using the Clevenger's apparatus. Then distillation carried out for 5 minutes utilizing the dry leaves and 1:10 water to leaf ratio. Anhydrous sodium sulfate was used to dehydrate the essential oil, which was then stored at a 4 degrees Celsius. Then phytoconstituent of oil isolated from the extracted oil. I have chosen the essential oil for the formulation of nanocapsule because essential oil have low stability. So by formulating the nanocapsule, by coating it by the polymer, we can improve its life. We can improve the stability of essential oil. Expected outcomes. This will meet the unmet need of stability enhancement of essential oil by decrease their volatility. Increase the oral compatibility. Then encapsulation of bioactive compounds represent a feasible and efficient approach to modulate drug release, protect from the interaction with the environment, enhance their bioactivity, reduce toxicity, and improve patient compliance and the convenience. Future benefits, opportunity, and challenges of nanocapsules. The nanocapsule have ability to deliver existing drug to their target. Nanocapsule should allow as much as 10,000-fold decrease in drug dosage, reducing the harmful side effect of drug using chemotherapy. Opportunity and challenges. There are many challenges that exist for developing a technique like architecture of biomimetic polymer, control of sensitive drug, function of active drug targeting, bioresponsive triggered system interacting with the smart delivery, carrier for advanced polymer for delivery of protein, drug delivery technique were developed to deliver or control the rate and amount of drug delivery. Finally, the nanocapsule can see novel effective drug delivery system in future. Thank you.
Thank you for your presentation, ma'am. Our respected session chairs, if you have any questions regarding the presentation, you can ask to the presenter.
No question.
Okay, thank you, ma'am, for your presentation. Thank you, everyone, for your presentation. I will read the names, the one who was not able to present before. I will call out the names. If you're available at the moment, let me know.
Good evening, ma'am. I am Dr. Ramani, ma'am. I am waiting for the presentation, ma'am.
Okay, yes, ma'am, you can.
Shall I start the screen share, ma'am?
Yes, ma'am, you can share it.
Thank you very much, ma'am. Thank you for the opportunity, ma'am. Thank you. Could you see this?
No, screen is not visible.
I am sharing my. Could you see this, ma'am?
No,
I'm sharing.
No. We can't see your screen.
1 minute, ma'am. 1 minute, ma'am. Give me 1 minute, ma'am.
Could you see this, ma'am? Now you can see this, ma'am. My chat.
Now
PPT
it's
sharing, ma'am.
you can continue.
Yes, ma'am. Thank you very much, ma'am. Good evening to all. I am Dr. M. Ramani, PhD Research Scholar, Department of Siddha, Tamil Nadu Dr. M.G. Medical University, Gindi, Chennai. My co-authors, Professor Dr. N. Kabilan, Professor and Head, Department of Siddha, Tamil Nadu Dr. M.G. Medical University, Gindi, Chennai, and co-author, Professor Dr. S. Chitra, Sairam Siddha Medical College, Chennai. My topic is molecular docking based screening of the constituents of Hyoscyamus niger identifies potential inhibitors of Alzheimer's disease, target acetylcholinesterase. Introduction: Alzheimer's disease is a neurodegenerative life-threatening disease which is caused by two proteins, namely beta amyloid and phosphorylated tau protein tangles. 50 million people are affected in worldwide. In India, there are 4 million are affected by the Alzheimer's disease. ApoE4 gene plays a major role in Alzheimer's disease. The brain starts to shrink. Later stages, this disease causes cognitive impairment, loss of memory, difficulty in learning, and brain cell death. In healthy brain, acetylcholinesterase is required in the breakdown of neurotransmitter acetylcholine. Aim and objective: the present study, we retrieved 6 bioactive compounds such as hyoscyamin, vanillin, belladonna, skimiamine, tropin, donepezil, which inhibit the target enzyme acetylcholinesterase, may act as a potential therapeutic agent for the management of Alzheimer's disease. Materials and methods: list of phytocomponents selected for docking in the herb Hyoscyamus niger seed, hyoscyamin, vanillin, belladonna, skimiamine, and tropin. This is the reference of the Hyoscyamin niger seed. Standard drug, donepezil. The lead molecule to bind with these core bioactive amino acid residue: tyrosine 72, leucine 76, tyrosine 77, proline 78, serine 347, and aspartate and glucine tyrosine, which mediates the enzymatic action of the acetylcholinesterase. The amino acid residues inhibit the enzymatic action of the cholinesterase, thereby restores the availability of the neurotransmitter called acetylcholine. Increased level of the acetylcholine improves the cognition and elevates memory retention potential by synergizing the neural communication. The lead molecules, phytochemicals that inhibit the enzyme acetylcholinesterase, will improve the brain level acetylcholine, considered as the best therapeutic choice for the management of Alzheimer's disease. This is the 3D structure of the acetylcholinesterase protein data bank 1B41. This is the receptor structure and crystalline structure of the target protein acetylcholinesterase with protein data bank 1B41 was retrieved from the protein data bank, and protein cleanup process was done, and essential missing hydrogen atom were being added. Different orientation of the lead molecules with respect to the target protein was evaluated by autodock program, and the best dock pose was selected based on the interaction study in analysis. Materials and methods: docking analysis calculations were carried out by using autodock 4. Gas TGR partial charges were added to the ligand atom. Non-polar hydrogen atom were merged. Rotatable bonds were defined, and docking calculations were carried out for the retrieved phytocomponents against the target protein. Essential hydrogen atom, Coleman united atom type charges and solvation parameters were added by the autodock tools. Affinity map 2x Armstrong grid point and 0.375 Armstrong spacing were generated using the autodock program. Autodock parameters set the distance dependent dielectric functions were used in the calculation of the van der Waals and the electrostatic terms respectively. Docking calculations were performed using the Lamarckian genetic algorithm and Solis as well as search method. Initial position, orientation, and torsion of the ligand molecules were set randomly. All rotatable torsion were released during docking, and each docking experiments were derived from 2 different runs that were set to terminate. After a maximum 250,000 energy evaluation, the population size was set to 150. During the search, the translational setup 0.2 Armstrong and quaternion and torsion steps 5 were applied. This is the 2D and 3D structures of the phytocomponents. Hyoscyamin, this is a 2D and 3D structure, and vanillin and beta-donine and skimiamine and tropin and donepezil. This is the ligand properties of the compound selected for docking against the acetylcholinesterase 1B41, the compound hyoscyamin, molecular weight gram per molecule 284.4 gram per molecule, vanillin 152.15 gram per molecule, and belladonna and skimianine, tropin, and donepezil. These are all in the tables. Summary of the molecular docking studies of the compounds against acetylcholinesterase, the compounds and binding free energy and inhibition constant and electrostatic energy, intermolecular energy, total interaction, all are given in the tables. Amino acid residue interactions of lead and standard against crystal structure of the acetylcholinesterase 1B41. Hyoscyamin gives two interactions with the amino acid residue, and vanillin 1 interaction with the amino acid residues, and belladonna 3 interactions with the amino acid residue, and tropin there is no amino acid residues, and donepezil 5 interactions amino acid residue. The results, total of 5 bioactive lead components were retrieved from the herb Hyoscyamus niger seed. From the reported data of the herb, based on the binding interaction analysis, it was identified that the chemicals hyoscyamin, vanillin, and belladonna reveals 1 to 3 significant interactions with the core target amino acid present on the protein acetylcholinesterase enzyme in comparison with the standard drug donepezil. And conclusion, based on the computational analysis, the bioactive compounds hyoscyamin, vanillin, and belladonna are present in the herb Hyoscyamus niger reveals significant binding against the target protein acetylcholinesterase by interacting with the amino acid present on the active site. Hence, these active phytocomponents inhibit the target protein acetylcholinesterase, may act as a potential therapeutic agent for the management of Alzheimer's disease. These are all the references. Thank you for your patient hearing. Thank you very much. Thank you for all guest lecturers. And once again, thank you very much, ma'am.
Thank you, ma'am. Thank you for your presentation. Dear session chairs, if you have any questions regarding the presentation, you can ask to the presenter.
Ma'am, you have mentioned 3 compounds are showing activity, inhibitory property.
Yes, ma'am. Hyoscyamin and vanillin and belladonna reveals 1 to 3 significant interactions.
Yeah.
Yes, ma'am.
And only this selected parameter is chosen, or
Yes,
any
ma'am. No. I have 5 bioactive components, but there is no interactions. Hyoscyamin and vanillin, belladonna give the interactions with the amino acid residues. Skimianine and tropin, there is no amino acid residues, ma'am.
Yeah.
Don't
And
the standard drug, ma'am.
binding ability?
Yes, ma'am. I have given in the table, ma'am, amino acid residues. Binding ability 2 interactions, ma'am. 72 tyrosine and 74 aspartate, 86 tryptonin, 87.
So
I have
again,
given in the table, ma'am.
Only having
Yes,
best
ma'am.
binding ability.
Yeah, yes, ma'am.
Okay, ma'am. Thank you. Thank you.
Thank you very much, ma'am. Thank you for your kind cooperation, ma'am. Thank you very much, ma'am.
Thank you, ma'am. Thank you for your presentation.
Thank you, ma'am.
This is who will do your presentation. Please be available. I will call your names. Let me know if you are available. A. Agalia from Government Arts College, Coimbatore. Are you available, ma'am? Are you there? Next, we move to our next presenter, Said Halim from Lemsen University, Algeria. Next, we'll call upon our presenter, Kritika K, SRM Institute of Science and Technology, Katankalathur, Chengalpattu. Next, we call upon our presenter, Sabha Manbili from Sri Ramachandra Faculty of Pharmacy. Next, we call upon our next presenter, Dr. C. Meenakshi, Government Siddha Medical College, Chennai. Next, we call upon our next presenter, Shalini B, National Institute of Siddha. Thank you all keynote speakers, session chairs, participants, presenters for joining today's session. I hope you have gained valuable insight from day one session. Looking forward to meet you all tomorrow. Please note the conference starts from tomorrow at 9:20 a.m. You can join the day's 2 session with the same link you used for today. Looking forward to your presence tomorrow. The certificate will be provided after 5 to 7 days once the conference is over. Thank you. Thank everyone here.